So good afternoon, everyone. My name is Talah Nammor, as you can guys see here. Today we're going to go over gastric secretion and filling and emptying. They're two different lectures. But I will start with gastric secretion, and then as we finish it, we're gonna go into the filling and emptying. I just want you guys to stop me whenever you have any questions, whenever you have any problem with something that I explained, please just tell me on the chat. I have the chat open, and let's start. I know it seems like this lecture is like شوي hard, but trust me. Wait, why is it? Okay, it's not that hard, inshallah. So, first of all, we know that we're talking about the stomach, okay? I am sure, guys, that in anatomy, you covered that the stomach is made up of many different parts, okay? We have the fundus, we have the body, we have the antrum, and then we have in here the pylorus, okay? Now, in physiology, we have functional divisions for those areas. Okay, functional divisions. Why do we have functional divisions? We have functional divisions because basically, as we all know, gastric secretions are released from glands. So we have two different glands in our stomach. We have the oxyntic gland and we have the pyloric gland. Therefore, we have two different areas in the stomach. We have this area, which is basically the fundus and the body up until here, which makes up literally 80% of the stomach, contains the oxyntic glands. So basically it's called the oxyntic gland area, okay? And then we have this 20% in here, which is the distal 20%, which contains the pyloric gland, okay? So this is really important because the oxyntic gland contains cells that are like different from the cells that are found in the pyloric gland, and each cell in the GI secretes different hormones. So it's really important, guys, to know that the proximal 80% is the oxyntic gland area, and the 20% is the pyloric gland area. Now, from the name oxyntic, okay, we know that basically, So we know that basically, what is it called? It produces acid, which is the gastric acid that we have in our stomach, which is the hydrogen chloride, okay? Now, the distal 20%, it is the part that produces the gastrin. And gastrin is a very, very important, what is it called? A very, very important hormone for the regulation of the secretion of the gastric acid, okay? Okay. Now, in the next slide, PowerPoint is acting up. Okay, in here, Dr. Peter included this slide to show you the two different glands, okay? So on our left side in here, we have the oxyntic gland, okay? And then in here, we have the pyloric gland. Both of them are made up of stem cells and surface mucus cells, okay? But the difference between the oxyntic gland and the pyloric gland, when we're talking about like the stem cells and the mucus cells, is that the stem cells in the oxyntic gland, they are found closer to the neck of the oxyntic gland, okay? While in the pyloric gland, they're more like down and they're closer to the isthmus, or basically, you know, the folds. Basically, isthmus means a fold. So the stem cell in here, it's closer to the fold down, while in the oxyntic gland, it's like right at the top and the neck, okay? Now, this is important just to differentiate between the two different glands. Yani, it helps us to identify, oh, okay, the stem cell is here, so this is the oxyntic gland. The stem cell is down, like further down, this is the pyloric gland, okay? And it's important, guys, to know that the stem cells are the basic cells that give rise to all the other cells, okay? So basically, they will give rise to all the different types of cells that we have in the oxyntic gland and in the pyloric gland. So they're very essential. And then you can see, guys, those like very normal looking cells, those are called, what is it called? The mucus cells. Now, the mucus cells are very important to release mucus. And this mucus protects our GI from the very harsh environment that we have in the GI. We all know that the GI is made up of, the GI contains hydrochloric acid, which is really, really acidic. And we don't want this hydrochloric acid to affect our GI mucosa. Therefore, we have a lot of mucus cells that will release mucus that will protect the GI from its own gastric acid secretions. Okay. It's very important to know that basically the mucus cells, they keep on like dying every 3 to 5 days. Like their lifespan is between 3 to 5 days. So it makes sense because, guys, if you can imagine the gastric like acid environment, like the gastric acid and the gastric environment in general, it's very harsh. So it will keep on like killing the cells. Okay. So sorry guys, I'm sick. You're gonna hear me cough a lot. Anyways, so basically, so it's important to know every 3 to 5 days, the mucus cells will die, and then they're gonna be shed out, and then the stem cells will give rise to new mucus cells. Okay, and then we have something called parietal and chief cells. I think, guys, you covered this. Do you know what the parietal and chief cells are?
Yes.
Okay, perfect. So basically, the parietal and the chief cells, they are only found in the oxyntic gland, okay? They're not as often found in the pyloric gland. And because they're very special cells, their like lifespan is way, way, way longer than the surface mucus cells, and they last from 90 to 120 days, okay? This is pretty much very easy slide. It's nothing hard, you guys know that. Yeah, sure. What do you want me to repeat? Like which point? I don't mind repeating. Okay, which gland secretes mucus? So basically, guys, both glands will secrete mucus because as you can see, both glands are made up of mucus cells, okay? Yeah, okay, sure. Okay, I'll start in here and then I'll go back, okay. So then we want me guys to repeat. So first of all, we were talking about what is it called? The stomach. The stomach has functional divisions. What do we mean by functional divisions? Basically, you all know the basic anatomy of the stomach, the fundus, the body, the antrum, the pylorus. This is not what we need here in physiology. In physiology, you guys have to know that the proximal 80%, which is basically the fundus, the body, and part of the antrum, is the oxyntic gland area, okay? The oxyntic gland area is the area that secretes the gastric acid, okay? Okay. But then we move on into the pylorus. In the pylorus, we have the pyloric gland. And it's kind of easy, pylorus, pyloric gland. So basically the pyloric gland will be in the pylorus and it is like responsible for the production of the gastrin, okay? Now, gastrin is a hormone that we're going to talk a lot about later on, but like as a simple function of gastrin, think of it as gastrin is one of the hormones that stimulates the release of the acid. And so you can see, guys, here there's an arrow between this and this. It's just that the doctor is trying to tell you that the gastrin production, okay, will result in the acid production. As we're going to go over this again, like later on in the slides, as the most important thing in this slide is for you guys to know that we have oxyntic gland area, which is the proximal 80, and distal 20 is the pyloric gland area. It's easy to memorize that the pyloric gland area is the distal 20 because of the position of the pylorus. Yani pyloric comes from pylorus. So since it's the pylorus is at the end, like at the distant end, so basically the pyloric gland will be at the distal 20%. Okay, guys, is this slide clear? Should I move on?
Yes, please.
So, in this slide, okay, we were saying, no, we have the two different glands that we mentioned before. So we said we have functional divisions. We said the proximal 80 is made up of the oxyntic gland. So okay, what is the oxyntic gland? We have the oxyntic gland here, okay? The oxyntic gland is the gland that is responsible for the production of the gastric acid. Hello? So basically, when we need gastric acid, we're going to find the cells that produce the gastric acid in here, in the oxyntic gland. The pyloric gland will not contain those cells that produce the gastric acid. Why? Because basically it is responsible for the production of gastrin. So it's going to contain cells that produce gastrin. Okay? We're going to explain those cells in the next slide. But the important thing in here is the important thing in here is to know that we have basically like the basic structure that both of them share. So both of them contain stem cells. Both of them contain mucus cells, okay? So the stem cells, they are basically the like mother of all cells. So whatever cell we need, the stem cell is going to differentiate and give us the cell, okay? So basically we have a stem cell in the axanthic gland and we have a stem cell in the pyloric gland. The difference between the stem cells in both of them is just the position. The position in the axanthic gland is closer to the neck of the axanthic gland. But in the pyloric gland, it's way deep down in the isthmus, okay? This is important, and it's just a point to differentiate for you. Yani if you see the stem cells down, just know this is pyloric. If you see the stem cell way up, this is axanthic, okay? Okay. And we have the mucus cells. Do you see, guys, those cells that are gray and they're like very average shaped? Like if you compare them to all the other cells, they look very boring. Those are the mucus cells, okay? The mucus cells are there to produce mucus, or the mucus that covers our stomach, okay? Now, it's important to know that those surface mucus cells are found in both. Mucus is secreted in both of them, okay? They're not in the surface, like they're just at the neck, you get me? Like way top, because this is the surface technically. So they're not literally on the surface, they're just at the neck of it, okay? Like once you go down, you're directly gonna find the stem cells. But you have to go deeper in the pyloric gland to find the stem cells. You get me? Someone asked in the chat. Anyways, so back to the surface mucus cells. We said the mucus cells, they're found in both glands. Why? Because guys, if you think of it, okay, let's go back here. The stomach is made up of both glands, okay? And in the pyloric area, we don't have axanthic glands. We only have pyloric glands. So we do need mucus cells in there to basically provide the protective sheet. Yani mucus, as we all know, it protects the stomach itself from its own acids, from its own enzymes, and so on. So basically, it will make sense, and we're gonna have mucus cells in both glands. Okay, is this point clear? Because it was mentioned earlier, which gland has the mucus cells.
Yes.
Yeah, you
Clear.
can think of that. True, the mucus cell is the blue layer in the diagram. Yani, basically, this mucus is the thing that protects the stomach. If you have to, yani, if you think of it, we know that the gastric acid is very, very, yani, it's very lethal to our mucosa. If it comes in contact with the mucosa, it's going to cause like damage. That's why we have our own protective layer, which is the mucus. So we have the mucus cells. Okay. And as I just mentioned, the acid is very, very like lethal, okay? That's why the mucus cell lifespan is very short. It's 3 to 5 days, okay? So basically every 3 to 5 days, the mucus cells are going to die. They're going to shed, okay? And then new mucus cells from the stem cells are going to like form so that they keep on reforming mucus cell, like mucus. Okay, guys? Is this clear?
Can I ask you a question?
Yeah, sure.
The pyloric gland, it has more surface mucus cells than the stem cells, than the axanthic gland. So the pyloric gland release more mucus than the axanthic gland, or it has nothing to do with.
I think
like,
listen, there is no, like the doctor didn't mention this anywhere, okay? Because both of them are both of them are the same, you get me? Like they're both are made of mucus cells. It's just that in here, guys, you see that the mucus cells are not as as often as here because we have more cells in the axanthic gland that we need to go over, okay? But it is not a rule. It's not like pyloric gland has more mucus cell, releases more mucus. No, خلاص, just know that it's made up of mucus cells and it produces mucus. Okay, don't go into such details that are not very necessary. And it's not they're not going to ask you which gland produces more mucus. Okay. Okay. So, moving on, someone asked what's the progenitor zone. The progenitor zone is just the zone where basically the stem cell, okay, you see the stem cell here, and then it will start to differentiate into something else. So the zone at which the differentiation happens is usually called the progenitor zone, okay? Okay, so someone asked, why does parietal and chief cells have a longer lifespan? Okay, so they have a longer lifespan because if you can see, guys, in here, they are at the lower end, okay? They're not as exposed to the gastric like secretions and to the enzymes and to all of the, what is it called? To all of the acid. So they're more protected. And I think, yani, God made them in a way that they can bear the environment way much compared to the surface mucus cells. So does that answer your question, or shall I say anything else? Okay. Moving on, we're going to go over the specific cells that are found in the oxyntic gland, and then we're going to go to the pyloric gland, okay? Try to ignore the right side. We're not going to talk about the pyloric gland for now. We're going to concentrate on the oxyntic, okay? So we discussed the mucus cell, we discussed the stem cell. Now we're going to the parietal cell. So can someone tell me, what does the parietal cell secrete?
HCl.
HCl, correct. Yeah. So basically, the parietal cells, they are the cells that are responsible for the release of the acid, okay? And they will release basically very important like molecules, which are called intrinsic factor. Does anyone know what is the intrinsic factor?
It's for the absorption of vitamin B12 in the intestines.
Excellent. So basically, yeah, basically the parietal cells, they will secrete intrinsic factor. Now, intrinsic factor is really important because it binds to vitamin B12. If we don't have intrinsic factor, our body is not capable of absorbing B12. So it's so guys, you just have to know parietal cell. What does it secrete? HCl. What does it secrete as well? Intrinsic factor. That's it. Okay. Tamam?
Yes.
Okay. Moving on, we have the interchromaffin-like cell. So the interchromaffin-like cell is a cell that is, like you can see, it's so small here, right? It releases histamine, okay? And histamine is a very, very important hormone. Why? Does anyone know why?
Because it's released for allergies.
That's correct. It is. It's usually for like inflammation and allergies, but it's not. This is not the way it works in the stomach. So
So
histamine is
basically,
what?
so basically, gastrin activates the ATC cells, so they can release. So what's it called? It's called serotonin. So
Oh.
so that you can activate the HCl.
Okay, you're kind of right, but it's not exactly this way because it's more it's okay, I'll tell you what, guys. Okay, so histamine is important because, as someone said, it triggers HCl secretion, okay? We said that the parietal cells, they release the gastric acid, right, which is HCl. طبعا HCl cannot be released unless there is like, you know, guys, the histamine gastrin axis. Do you guys remember this axis? If you don't, خلاص, we're just going to talk about it now. So basically, for the parietal cells to secrete HCl, it requires the interchromaffin-like cells to release histamine. So interchromaffin-like cells, they release histamine, and this histamine will travel all the way up into the parietal cells, and it will bind to receptors that is like specifically there to like bind to histamine, and then this will bring about a change. This change will result in the formation of HCl. Okay, we're going to go into like the details of this later on, but it's important, guys, to know that interchromaffin-like cells, they're very important due to the release of histamine. And why is histamine important? I just mentioned.
To release HCl.
Okay, perfect. Yeah, exactly. Histamine will bind to parietal cells. And I'll show you, inshallah, in the like forward slides, you're gonna see how does this exactly happen. Okay, so we finished from the interchromaffin-like cell. Guys, listen, I'll tell you something for this slide, okay? You don't really need to know how do they look like. You just have to know, guys, in a parietal cell, okay, مثلا, you have to know the name of the cell. We have parietal cell. And then, okay, what does it secrete? It secretes, مثلا, this hormone. Okay, what does this hormone do? That's what you guys have to know. That's it. And then later on, مثلا, you learn a little bit more details. As for this slide, the only important thing is to know that, okay, we have oxyntic gland. In the oxyntic gland, what are the cells there? Okay, and then we go over them. Like, okay, we have parietal cell. Okay, and then we have interchromaffin-like cell. Okay, so is this clear? Come on. So, guys, do you need to repeat anything about parietal cell or interchromaffin-like cell? Oh, perfect. Okay, moving on, we have the D cell. The D cell is not a specific cell that is only found in the oxyntic gland. As you guys can see here, we have a D cell in here too, okay? Listen, I took the same slides. This is literally your slides, but I removed what the doctor, like sometimes the doctor wrote stuff like that. What does paracrine mean? Paracrine means that it secretes hormones, okay? So basically, both glands are paracrine glands because both of them secrete hormones. You get me? Does this answer your question? Because guys, someone in the chat asked, why does it say paracrine in our slides? So paracrine, it just means that a gland secretes hormones. So as you can see, those different cells, they release hormones. So both of them are like supposedly to be paracrines, like that. Okay. Now moving on, as I'm saying, D cell. So the D cell is not specific to the oxyntic gland. It's found as well in the pyloric gland, and it releases a very important hormone, which is the somatostatin. Okay. The somatostatin hormone is very important because it is the only inhibitory hormone in this like in those cells. Okay, so the what is it called, the D cell? Yeah, don't worry, I'm gonna cover everything. Okay, what is it called? It will reduce HCl. Excellent. So basically the D cell is the only inhibitory hormone. Okay, it releases some D cell will release the only inhibitory hormone, which is the somatostatin. So this somatostatin, it's like the big boss. Okay, it keeps everything in check. So if we have, مثلا, if you think of it, if we're not eating anything, why would we need the gastric acid secretion? We wouldn't need it because there's no food. It's like useless to release acid. So what happens is that the D cell will know that, oh, okay, there is no food. We don't need the gastric acid secretions. So it will release somatostatin. And somatostatin is an inhibitory hormone that will inhibit everything, okay? It will inhibit the release of HCl. It will inhibit other like things that happen too. But most importantly, know that it will inhibit the release of HCl. Okay, guys? Is this clear? Okay. Moving on, we have the chief cells. Okay, so the chief cells are, as again, only found in the oxyntic gland. The chief cells, they release pepsinogen. Pepsinogen is basically an enzyme. This enzyme, it is the inactive form of pepsin. And pepsin is an enzyme that breaks down, what is it called? The proteins. It breaks down the proteins from like long chain peptides into tinier amino acids, okay? So that's mainly what the chief cell secretes, but it also secretes gastric lipase, and it's another enzyme, that breaks down lipids into, basically, it breaks down triglycerides into basically monoglycerides and fatty acids. Okay, so just know that chief cell is like, it's like a vesicle that has a lot of enzymes. Okay, sure. So the chief cells, it's just a cell that contains, like, you can think of it, it contains most of the enzymes. Okay, so once the chief cells, they're activated, they will release two enzymes. What are they? They're pepsinogen and lipase, gastric lipase. So pepsinogen, okay, pepsinogen is the inactive form of pepsin. And pepsin is an enzyme that breaks down the polypeptides, which is basically the proteins, into smaller amino acids. Okay? Is this clear? For pepsinogen, and then we have basically the lipase. The gastric lipase will basically break down the long chains fatty acids into smaller chains into fatty acids and like free fatty acids and like monoglycerides. Okay? Okay. Last but not least, we have the interchromaffin cells. Interchromaffin cells are just like the D cells. They're found on both of them. Okay? So they're found in the axanthic gland and the pyloric gland. So they're not specific for the axanthic gland. Interchromaffin cell, they release serotonin, okay? And serotonin is just basically a hormone that makes, it activates basically our enteric nervous system, and it brings about the secretion of many of those hormones that we already have. And I'm gonna go over this in the next slide, okay? Is this clear?
Yes.
Okay.
Just one last thing. So basically, the G cells send signals to the ECT cells, so they can release histamine. So parietal cells can release HCl.
Yeah.
And so the chief cells release pepsinogen, so the parietal cells can transform that to pepsin, so we can digest food. But what's the use of HCl?
What's the use of HCl? Okay, that's an amazing question. So basically, the main use of HCl, first of all, it's to protect our body from all the bacteria that can come when you ingest the food. Okay, that's one of the most important functions of the HCl. So basically, because it provides such an acidic environment, the bacteria and all of those pathogens that come in the food, they cannot survive. Another very important function of the HCl is that it, what is it called? It provides the perfect, like the optimum pH for a lot of the enzymes, for example, pepsinogen, to undergo the transformational change into the active form, which is pepsin. Okay. As well as the last thing, it helps in the denaturing of the proteins. So it's, if you can think of it, and it protects our body from bacteria. It makes it easier for our body to digest the food by activating the enzymes, and it makes it easier for our body to digest the food by breaking the proteins. Like the protein you eat, it's going to become denatured, so it's easier for the enzymes to work on it. Okay.
Can you repeat what's the interchromaffin cells? Like, what does the sternum do?
Yeah, sure. So basically, the interchromaffin cell will release serotonin. Okay. No, I didn't yet explain G cells. Don't worry. Because we're just gonna finish axanthic gland and move on to pyloric glands. Okay. Anyways, for interchromaffin cell, they release serotonin. Okay. Serotonin is a hormone that activates the nerves. Okay. It can either activate the nerve, or it can basically act. You know how basically, guys, do you remember in the lecture Ghaid explained yesterday? Do you remember how we have in the autonomic nervous system, we have stimulatory neurons and inhibitory neurons?
Yes.
Yes. So basically, serotonin, it will bind to the one of those neurons, depending on the situation. So if, مثلا, we want the vagovagal reflex to work, it will bind to the, basically, what is it called? It will go and bind to the CNS, and it will result in the release of acetylcholine. You get me? So basically, serotonin is part of our enteric nervous system. Okay. Is it clear? We're gonna talk about.
Yeah.
Yes.
So serotonin is basically a hormone that activates the neurons that are needed for the breaking down.
Yeah, listen, we're going to go exactly over the function later on. Let me not confuse you from now. بس نروح the slide, I'll explain it, okay? Is this okay with you guys?
Yes, I understand.
Yeah. Because now it's gonna seem like I'm just throwing words at you guys, but later on, Okay, it's okay. Serotonin, it works on the nervous system. Okay, so it either activates it activates a stimulatory neuron or it activates an inhibitory neuron depending on the situation. Is it clear? Okay. So basically, guys, this is everything for axanthic gland, like all the cells that are there. Do you want me to repeat any of the cells before I move on to the pyloric gland? Okay, perfect. So basically, with pyloric gland, we only have one cell that is different than the axanthic gland, and it's a very, very special cell. It's called the G cell. Okay, and guys, it's pretty clear. Yani, when we see G cell, so something that starts with G, so it will release gastrin. In here they say gastric. Ignore this. This is wrong. It's gastrin. Okay. Now, gastrin is a very important hormone. Why? Because as I mentioned earlier, to release HCl, we need the gastrin histamine axis. Okay. So if we go back, wait, I want to show you something. Wait. Okay, so if we go back in here, in the pyloric gland or in the pylorus, in here, we're gonna have G cells. Those G cells, they will produce gastrin, okay? This gastrin will go all the way up into the proximal area where we have the axanthic gland, okay? And then it will go all the way up and then it will bind to what? Into interchromaffin-like cell, which is the cell that releases histamine. Once the gastrin binds to interchromaffin-like cell, okay, what happens? It will bring about the release of histamine. So histamine is going to be released, and this histamine will find its way to the parietal cell, bind to the parietal cell, and cause the release of basically HCl.
I'm kind of confused. So basically the G cells are, you said that the gastrin basically it acts as a regulator of the release of HCl. So basically what the gastric do is that it activates the ECL, and the ECL will release histamine so that it will release which activates the parietal cell. So like basically it's a pathway,
Yeah.
right?
Yeah, it's a pathway, exactly.
So it does not the G cells do not inhibit anything. They activate.
Gastrin, the G cells, yeah, they activate.
Okay, thank you.
Okay, so guys, as you can see here, this is the gastrin histamine axis that I was explaining earlier. So from the, what is it called? From the pyloric gland, the G cells in here, they will release gastrin. This gastrin will go up. It can go through the blood or it can go through through the stomach itself, but yeah, it makes more sense to go through blood, okay? So it will go through blood all the way into ECL cells, which are found where? The axanthic gland. So they're gonna bind to them. The axanthic glands are gonna release histamine, which is gonna go to the HCl and result in the formation of HCl. Okay? Is this clear, guys? This is a very important concept. But if you have any questions, please ask me because we're slowly building the information, okay? Okay, what does it mean? It's just that they like to use fancy words. So gastrin-histamine axis means that the amount of gastrin released compared to the amount of histamine. So if those both are increased, this has to mean this has to bring about the release of HCl. You get me? Like it's just the fancy name of this whole pathway. Okay. Okay. Guys, is it this clear, please? This is a very important slide.
Yes, it's clear.
خلاص, I'll move on.
Thank you.
تمام. it's linked exactly. The axanthic gland is linked to the pyloric gland by the G cell process. Yeah, like they're not actually linked. No, it's just that you guys have to know that the axanthic gland have the parietal cells. The parietal cells will depend on the G cells that are on the pyloric gland. Okay? Okay. And someone asked, does the HCl activate the pepsinogen? Yep, the HCl will activate the pepsinogen. Now this slide, it's just a summary of everything I told you guys. So as we mentioned, we have parietal cells. They will release intrinsic factor. They will release hydrochloric acid, which is HCl. And then we said the function of the intrinsic factor is to bind to vitamin B. And it's very important because vitamin B is not going to be absorbed in the ileum without the intrinsic factor, okay? And then we said the function of the hydrochloric acid is to denature the protein to, what is it called? To prevent, impedes bacterial colonization is basically prevent the growth of bacteria, okay? So it will prevent the growth of the bacteria by killing it. Bactericidal means it's gonna kill the bacteria, or bacteriostatic means that it's gonna inhibit the reproduction of it. So we're gonna have the bacteria, but it's not gonna reproduce, so it's not gonna cause an infection, okay? And then it converts pepsinogen to pepsin. Someone in the chat asked, what stimulates the release of pepsinogen and lipase from the chief cell? I'm gonna explain that in the in the following slides. So just bear with me, guys. And then we have basically the chief cells. It releases pepsin, which is basically the pepsinogen, but it's activated, and gastric lipases, and it causes the formation of diglycerides and fatty acids, and here it causes the formation of amino acids. Is this clear? I think it is, right? Yeah. Last but not least, we said we have, yeah, ask. Someone has a question. Chief cells, pepsin activates HCl in parietal. No, it's not that way. It's the opposite. The HCl in the parietal will cause the pepsinogen. Oh, okay. Yeah. It will cause the pepsinogen to be activated into pepsin. Yeah. Okay. And then last but not least, guys, mucus cells, they're the protective layer and they prevent bacterial formation, and they lubricate. Okay, that's it for this slide. It's easy. You guys can just read it, memorize it. Yeah. Now we're going, guys, now to discuss the parietal self, like the parietal cell itself, and the secretion of HCl. So, first of all, can you guys tell me what do you see in this parietal cell? Anyone? Just anything you see. Yeah, mitochondria, exactly. Okay, so we have a lot of structures that make up the parietal cell. But yeah, as you can see here, we have a lot of mitochondria. And what does mitochondria produce? Yellow. That's a very easy question. Exactly, ATP.
ATP.
Yes, ATP is the energy of the cell. So since we have a lot of mitochondria, yani it's it's common sense to know that basically it's the action of releasing gastric acid requires a lot of energy. Okay? Okay. And then if you look here, guys, we have those weird stuff that look like tubules. And then we have a lot of circles, and those circles contain a lot of, you can see inclusions and stuff inside. So basically, these are the tubulovesicular membranes, okay? And we can see that most of the parietal cell is made up of those tubulovesicular membranes. And last but not least, we can see the caniculi, okay? So why are these important? I'll tell you why. So let's say we ate something, and then our gastric, our stomach is yalla, I need to release gastric acid. Do you think if we release gastric acid from this resting parietal cell, will it produce a lot of gastric acid? يعني, do you think that this exactly no? And why will it not produce a lot of gastric acid? Because if you look at the surface area, أصلاً, when we produce gastric acid, we need a lot of space, and then we need basically a huge surface area to secrete the thing. But if we look at this one, it's not gonna like produce a huge amounts of gastric acid, and that's why the first step that happens before the gastric acid secretion begins is that the parietal cell will undergo a conformational transformation. Okay, and then wait. Okay, and then what happens is that the tubulovesicular membranes that we see here, they will bind to the canaliculus, and they will result in the formation of those finger-like projections. Can you see them? There are like a lot of finger-like projections. Yeah, sure. Yeah, so someone asked me to repeat the tubulovesicular membrane. So the tubulovesicular membranes, they're just in here. You see, guys, tubulo, which is the tubules, these, like the lines, and then the vesicles are those circles. Vesicles, usually they are like spaces that contain stuff. So you can see a circle with stuff inside it, okay? Come on. So basically, those vesicles, they have a lot of dots in them. So those are the vesicles. And then we have the tubules. Those are the tubules. So together, there's tubulovesicular membrane, okay? Okay. And then the canaliculus in here, wait, this is the canaliculus. Guys, you don't have to know anything. You don't have to know exactly what's the function of these. You don't have to know anything. Just know parietal cell contains tubulovesicular membranes and canaliculus. Okay, and it contains a lot of mitochondria. That's it. Okay, when the resting parietal cell, or when the parietal cell needs to start working and start secreting HCl, a change will happen, a conformational change. And conformational change means that the shape of the cell will change. How will the shape of the cell change? What happens is those canaliculus, they will bind to the tubulovesicular membranes, resulting in the formation of those finger-like projections, which are called the microvilli. Okay, is this clear?
Yes.
Okay, now, guys, whenever you see microvilli, what's the first thing that comes to your mind? Absorption. Okay. But why does this absorption happen, in your opinion? Exactly, surface area. So basically, those microvilli are there to increase the surface area. So now, if you look at the structure of this parietal cell, we have a lot of space to secrete a lot of gastric acid. And we said earlier, and no, here the surface area is so tiny, it's not going to produce a lot. That's why it undergoes this change. So we have in a lot of surface area, so it secretes a lot of gastric acid. Okay. So for the parietal cell, just remember, for the parietal cell to start secreting HCl, it will go a change in its shape and its structure. How is this change? How does this change happen? It's just the tubulovesicular membranes will bind to the canaliculus. Yeah, gastric acid is HCl. Okay. No, it occurs every single time. So basically, we eat something now. It requires the release of HCl or gastric acid. So it will go to the tubulovesicular membranes and the canaliculus will fuse, and then they'll cause the formation of this. After this process is done, خلاص, gastric acid is secreted. They did their work. They're gonna go back to the resting stage and wait until مثلا another stimulus happens. Okay. Okay. Now, next. Okay, sorry. Now, guys, we mentioned all those different cells, and we talked about we talked about the gastrin histamine axis and so on. here is when like everything actually starts. Here we're going to discuss the cellular, okay? يعني how does it exactly happen, okay? Okay, so guys, I told you before, no, we need two hormones to secrete HCl. What are they?
Gastrin and histamine.
Excellent. So basically, as you guys can see here, we have histamine, we have gastrin, and surprisingly, we have ACH. You guys know acetylcholine, right? Kid, you know what's name. Just finished MSK. Anyways, so what you have to know, guys, is that this structure in here is the parietal cell, okay? At the base of the parietal cell, we have receptors, okay? Those receptors are waiting for their hormones. Yani we know for any hormone, it needs a receptor. So basically, at the base of this, we have the receptors, and they're waiting for the hormones to come to cause the change, okay? I'll take it step by step. I'll start with histamine, and then we can go to gastrin and acetylcholine. But I want you to know, guys, that all of those three hormones cause the release of HCl. But histamine is the strongest of them all. What does this mean? This means that we only have histamine. If we don't have gastrin, we don't have acetylcholine, we're going to secrete HCl because it's the strongest, okay? And as you can see here, I mentioned this for you. Histamine is the strongest, followed by acetylcholine, followed by gastrin. So if gastrin is there alone, it's not going to cause gastric acid secretion as much as if histamine is there, okay? Is this point clear? Yeah, perfect. So now we're going to talk about the receptors. So we have histamine. Okay, someone asked, gastrin causes histamine secretion. So how can histamine stimulate HCl secretion without gastrin? Akid, it's not going to stimulate, what you're saying is correct. But what I meant is that sometimes, if you think of it, if we go back, wait, show you. If we go back here, if this is the G cell, it releases gastrin. If the gastrin goes all the way into the interchromaffin-like cell, it might be خلاص consumed, and all the histamine will go to the parietal cells. You get me? This is my point. Gastrin will definitely has to be there, but it doesn't really have to be there in the parietal cell. It has to be there for the interchromaffin-like cell. Okay? Is this point clear, or did I confuse you guys?
I got a little bit confused.
Okay, listen. Now we have three hormones that bring about the release of HCl. But the most important one is histamine, and the strongest one is histamine, followed by acetylcholine, followed by gastrin. What I was saying, G cells, they will release gastrin. It depends on the amount of gastrin that is released. So this gastrin will go to the interchromaffin-like cells to cause the release of histamine. Well, this histamine will go to the parietal cell. If we have excess gastrin will go to the parietal cell and will cause the command secretion. But the main hormone is histamine, okay?
So like extra, so basically extra gastrin that is getting released, it goes to the parietal cell. But the most important function of the G cells or like the gastrin is to activate histamine.
Exactly. That's the main job is of okay, when you think of activation of HCl and activation of parietal cell, first hormone that comes to mind is histamine, okay? And gastrin's main function is to cause the release of histamine, okay?
Yes.
Tamam, guys? خلاص. Let's talk. Wait, so gastric can bind to parietal cell if there is an excess of it. You can put listen, guys. I'm telling you in يعني yes, it can bind to parietal cell if there is excess of it. Or كمان it can bind to يعني just know that we have what is it called? Just know that we have receptors for gastrin, and gastrin can cause gastric secretion without histamine, but it's very very weak. يعني إذا نحن we're gonna think of the power of the hormone, just think إنه histamine is the most powerful and gastrin is the weakest. Okay? تمام? Is this point clear? يعني I don't want you guys to think إنه gastrin cannot cause HCl secretion. No, it can cause HCl secretion, but it's not strong. يعني it's very weak. And what will يعني explain this more, when I go through the cellular pathway in here, you're going to see that basically gastrin alone is not as strong as histamine alone. Okay? Okay. Let's go through this like part here. So histamine will bind to a receptor called H2. It's pretty easy, guys. يعني ما فيها شيء. Histamine starts with an H. The receptor starts with an H, so H2. Okay. It's really important to remember the receptor names. I know it's a lot of receptors, but guys, it's easy if you think of it logically. So histamine will bind to H2. And then we have acetylcholine. Acetylcholine, it will bind to M3. طبعا why M3? Because we know that basically in the autonomic nervous system, or anything that is not voluntarily controlled by us, we have muscarinic cholinergic receptors, right? Do you remember this from Oscar? Yeah, exactly. So basically, muscarinic starts with an M, so acetylcholine will bind to M3. Okay, okay. I'm sorry. Someone said that me mentioning MSK gives them scary vibes. I'm sorry, guys. So what is it called? Moving on to the last one, gastrin. Gastrin will bind to CCK2. هلا CCK2 is a bit hard to memorize, so I made a mnemonic that is كمان شوي silly, but I feel like it does the job. So if you think of it, gastrin can cause secretion too. So if you take the first letter of every word, gastric, so gastrin, so gastrin itself, can, C, cause, C, secretion, I wrote the secretion with a K, so you guys can remember. So K, and then 2, because it's like an emphasis, like, oh yeah, it can secrete, it can cause secretion too, but gastrin equals CCK2. Is this clear? I know it's a very silly mnemonic, but yeah. المهم, now, thank you. Whoever said nice mnemonic, thank you guys. Anyways, histamine, I'll discuss the histamine pathway بعدين بنروح على الباقي. So, histamine will come. It will come from where? Guys, you have to tell me what cell secretes histamine. يلا بسرعة.
ECL?
Exactly. So ECL, it releases histamine. Histamine will bind to H2. طيب, we know when a hormone binds to a receptor, something has to be released. يعني they're not just gonna bind وخلاص. لا. So this hormone will bind to H2 and it will cause an increase in CAMP. Okay? So histamine binding to H2 will cause an increase in cyclic AMP. But why is this increase important? Okay, خلاص, CAMP is increased. Why is it important? It's important because cyclic AMP, you can think of it, it's like a form of energy. Yep. بس يعني, don't take my words literally. But think of it, CAMP, it will go, okay, it will go all the way up into this hydrogen potassium ATPase, and it will activate it, okay, and then it will go all the way into this potassium ion channel. It will activate it. It will go to the Cl ion channel. It will activate. Oh yeah. So okay, it's activated. What will happen? So in in the hydrogen potassium ATPase, okay, what will happen? Hydrogen will be secreted out. You can see, خلاص, hydrogen is secreted out, and then they take in a potassium. So hydrogen out, potassium in. Okay, and then guys, think of it. What is HCl made up of?
Hydrochloric
Hydrogen
acid.
chloride.
Yeah, so what is it made up of? Like the ion itself.
Hydrogen
H
chloride.
ions.
Exactly. So it's made up of hydrogen and it's made up of chloride. So the whole process needs to secrete hydrogen and chloride, and this is how it secretes it. So basically, it will push a hydrogen out and will push a potassium in. The potassium in is just where, you know, like this ATPase requires potassium in to release the hydrogen. But potassium goes in, hydrogen goes out. And then the cyclic AMP will go all the way into the Cl, the chloride channel, and then exactly. No, it's not exactly like it. It's just another type of pump. But يعني it's the same idea. The same idea of NAK pump, but it's made up of different ions. Anyways, the cyclic AMP, yeah, it's one pump for both. Yani, guys, look, this is the pump, okay? It requires one to go out, heck, one to go out like this, and one to go in like this. Can you see it? So basically, what goes out? Hydrogen, because we need the hydrogen out. So so for the hydrogen to go out, it needs something to go in of طبعا the same charge. So what goes in? Potassium. Okay, it's just guys know no hydrogen potassium ATPase pumps hydrogen out, potassium in. No, someone asked, is the hydrogen potassium ATPase only activated by the combination of both? No, this arrow combines with this just to tell you guys that if يعني إنه both of them act on HK ATPase. Okay, is this clear لهلا? Did you get how does it work? Perfect. طيب, so guys, this it's very important to know name of the receptor, name of the hormone, what does it cause increase in? يعني خلاص, histamine H binds to hydrogen. Sorry, not hydrogen. So histamine binds to H2, increases CAMP. CAMP will go exactly. It will trigger. It will work on the ATPase and يلا work. So it's gonna push the hydrogen out, push the potassium in, and then it will go and work on the Cl, push the Cl out. طب خلاص صار عندنا H, صار عندنا Cl. They will go, they'll meet to form HCl. Okay? Even if, wait. What do you mean two different channels? You mean the K? Okay, so, and I didn't want to talk about the K now to not confuse you, but guys, this K is just reduce, reuse, recycle. شي يعني reduce, reuse, recycle. So basically, the K goes in. نحن we don't really need the K in. And if it keeps on going in, okay, you know how basically the charge of the cell will go up, but hydrogen will not be able to go out. What happens? This K channel is just there to kick it out. So بصير في عنا مثل a circular motion. You see? It goes in, then goes out, then goes in, then goes out. You get me? It's just recycled so that the process can still go on without affecting the total charge على تبع الcell. Okay? Well, oh wait, there's a question. Even if cyclic AMP is inactivated by hay, then the calcium can still activate hydrogen K ATPase though. Yes, calcium can still activate the HK ATPase. But I'm gonna go over this later, guys. Like just after I explain the other two receptors, don't confuse yourself. And yeah, come on. Is everything till now clear? Does K plus activate Cl? No, K plus has to do nothing. Listen, guys. K, like potassium, has nothing to do with what does it activate. It's just a passenger, you know, like this what is it called? This this proton pump is just like a cab. It needs one person to go in after the other goes out. So basically, what goes in? The K. Yeah, sure. I knew. Okay, listen. I'll go over it again from the beginning. Cyclic AMP will increase. حلو. Cyclic AMP
لا لا
will
أنا
go.
هاي I think I understand it، بس أنا اللي قصدي بالكيل بامب هون. شوفي مش صح نحن بنطلع اتش لبرا وندخل كي لجوا.
Yeah.
ايش بصير بعدين؟
ايش بصير بعدين؟ هاي الكي بتكون هون. The K is here. It's gonna be recycled to go in again. You get me? It's like a circle. It goes out. Then
Yeah,
goes
because
in.
we cannot release hydrogen. We cannot release hydrogen without making. يعني hydrogen cannot be released without the K being inside or
Exactly.
the potassium, right?
Yeah.
So
I will.
we
yeah.
need the potassium. We need the potassium to be recycled so that we can release more hydrogen.
Yeah. Don't worry. I'll repeat it again. And yes, what you're saying is correct. CAMP. I'll repeat the histamine one more time, and then I'll move on to the gastrin and ACH. Okay. So histamine will bind to H2. This binding will cause an increase in cyclic AMP. This cyclic AMP, just think and cyclic AMP is the know-it-all. خلاص, and I can work on all of them. So what are the ion channels, or what are the things that we have in here? We have the proton pump اللي هي hydrogen, potassium, ATPase. We have the potassium ion channel. We have the Cl ion channel. Cyclic AMP will just go and work on them all. So it will go بتروح على H, the hydrogen K ATPase. It will activate it. Hydrogen is kicked out. Potassium is kicked in. طب okay, بس نحن we don't really need the potassium in. So the cyclic AMP gets us covered بتروح عند the potassium ion channel to kick the potassium out because we don't really need it in. Like now, يعني we don't really need it in. We need it out, so it goes in again. Get me? Like we don't really need the potassium to stay in. If it stays in, the hydrogen is not going to be released. So it will just go through this channel out. بعدين the cyclic AMP will go to the Cl, the Cl channel. This Cl channel will kick Cl out. So it just causes all of the ion channels to work. It causes them to release the ions that they hate. يعني مثلا hydrogen potassium ATPase will release hydrogen. The potassium ion channel will release potassium, and it will let it go out, and the Cl channel will let Cl go out. Is this clear?
Yes.
تمام. Do you want me to confuse you? Someone asked about the concentration of potassium. You can think of it like that, but I'll be honest with you, ATPases, they act against the concentration gradient. That's why they require ATP. But don't think of the concentration gradient. Don't confuse yourself, and don't confuse yourself or others. It's really going to be confusing for you to know it. خلاص, just know potassium in, بعد ما potassium بتفوت لجوا, it's going to be recycled out, and then it's just a process. Okay? But to answer your question, no, it's not down its concentration gradient. Moving on. Yep, exactly. That's why we have a lot of mitochondria, because we need a lot of ATP, because we have this ATPase, and this ATPase requires ATP. Perfect, guys. You're linking things together. Moving on, we have the ACH in gastrin. They will bind to their receptors. And then what happens is that they will cause the release of calcium. Tamam? But the thing is, calcium is not as strong as cyclic AMP. It will only act on one, what is it called? On one channel, which is this, the hydrogen potassium ATPase. Okay? So ACH will bind to M3, gastrin will bind to CCK2, and they will cause the release of calcium. So when this increased calcium levels, they will not work on any other channel except one, اللي هي ATPase. Okay? Tamam, guys? Remember how in the beginning I told you that histamine is the strongest of them all? This is why, because histamine will release CAMP, and a binding of histamine to H2 will result in the release of CAMP. And CAMP can work on all of the ion channels. Like you can see here, guys, it can work on this, it can work on that, and on this. But it can bring about the release of H and Cl minus. But gastrin and ACH, they will result in the increase of calcium, which will only okay, only act on the ATPase. Okay, is this clear? Ca can only activate the release of hydrogen, however, it cannot activate Cl minus. Yeah, exactly. So it cannot cause the release of Cl minus because it doesn't work on it. Okay, guys, this is a very important slide. Yani, if you understand this, you understand everything. Is this okay? Wait, how would HCl be produced if Cl is not being released? Exactly. Cl minus is not released. So basically, that's why gastrin alone cannot bring yani, the production of HCl as histamine. You get me? Like, this is what I was trying to go over in the beginning. That's why histamine is the strongest. But gastrin by itself, yani, listen, if gastrin was lucky, if gastrin was lucky and Cl minus was by luck there, okay, then the release of H alone will result in the formation of gastric acid. But if إذا نحن ما عندنا histamine, we don't have histamine, and only gastrin is there, يعني, unless we depend on luck, there's not going to be HCl production. Okay? You're welcome, guys. Anyways, so one last thing in this slide. Do you remember the big boss? Who is the big boss? No, histamine is not this big boss. كان في big boss تاني that keeps everything in check. SST صح. Somatostatin. So somatostatin, yeah, yeah, somatostatin. So basically SST, تمام. SST is the big boss. It keeps everything in check. So let's say we're not eating. Wait, someone's asking me to repeat. Repeat what? Everything or just big boss? After histamine. You mean the gastrin and ACH? Okay. Let's go back to that. So, guys, we have ACH, we have gastrin. ACH will bind to M3. Once it binds to M3, it will cause an increase in calcium levels. يعني كيف histamine causes increase in cyclic AMP? In here, acetylcholine will cause an increase in calcium levels. حلو? Gastrin كمان, when it binds to CCK2, it will cause an increase in calcium too. Calcium also, okay? Now, this calcium here, it will only work on one proton pump, which is this pump, the hydrogen potassium ATPase. It's not going to work on any other one. Also, you can see it here. It has only one arrow in here. It will not work on this. It will not work on this. It will only work on this. Okay? Is it clear? Like till now? Tamam. So since it will work on this, it will cause the release of hydrogen. So it will only cause the release of hydrogen. It cannot cause the release of Cl minus. That's why we were saying gastrin is the weakest, because basically gastrin will only bring about the release of hydrogen. And if we were what is it called? And if we were lucky, we have Cl هيك moving on, like moving there, it's gonna form HCl. But alone, gastrin alone is not gonna be able to produce that huge amounts of gastric acid. Okay? مين كاتب أخاف أنسى تنسي هالأشياء؟ والله it's easy. If you keep on repeating them, يعني ما بتحسي إنه بتنسيهم. Just use my mnemonic, guys. My mnemonic is amazing. Thank you, guys. Anyways, نرجع للbig boss situation. So let's say we're not eating, we're not خلاص يعني we don't need شو اسمه we don't need to, what is it called? We don't need acids, okay? So what happens? بيجي the big boss somatostatin and it binds to its receptor. طبعا the somatostatin receptor is so easy to remember. Why? Because SST, somatostatin, SST, and then you just add an R. R stands for receptor. So somatostatin receptor, okay? So somatostatin will bind to SSTR2, and then it will cause the decrease of CAMP. Are you sure? Because I know that the doctor said don't remember PGE, the prostaglandin. Prostaglandin is not important. But what is important, this is really important. Okay? Yeah, I know, but yeah. Don't worry, guys. I went over the slides يعني, with every single detail. قرأت all the notes. Anyways, this is important, but prostaglandins, ignore this. This is non-existent. Why is SSTR2 not SSRT? Well, I don't know, guys. It's just like that. Just know that it's SSTR2, okay? So what happens? بعد ما somatostatin binds to the receptor. يعني it's just a name, guys. يعني I don't want to افتي من عندي, I'm sorry. فبعد ما it binds to it, the cyclic AMP will decrease. Once cyclic AMP decreases, the ion channels خلاص, they will go to sleep. So basically, we're not going to have gastric acid secretion. Okay? Tamam? Is this point clear? This is a very important point, guys. Tamam? خلاص يلا, I'll move on. Your last chance to ask questions about this.
I think everything's great.
Perfect. Okay. هلا تتذكروا كيف كثير قعدنا نحكي عن هرمونز؟ هلا بدنا نحكي عن ions. Okay, عنا hydrogen وعنا chloride. طب from where are those coming? يعني okay, we have hydrogen, we have chloride. But if you think of it, the cell will not have just take randomly hydrogen جاي من هون and chloride جاي من هون. No. So what happens is, guys, this amazing photo. Look, like zoom in. What is the most common thing in our body? Our body is made of 80% of what?
Water.
Water. صح. So this is, as you can see, we have water here. Okay? طيب, ونحن, we know that our body is always active. Our metabolism is always working. So what is the most common metabolic waste? Yeah, sure, I can repeat. So listen. No. Okay, listen. أول شيء نحن, to form HCl, we said we need hydrogen and chloride. Okay. This slide is there to explain where do these come from. يعني نحن, we're not going to have them in the cell heck just lying there. No. They have to come from a place. Okay. So I asked them, what are we made up most from? And they said water. And then what is the most common metabolic waste? Exactly, that's carbon dioxide. So as you can see here, the first step in the formation of hydrogen is we're gonna have water, which is here, and then we're gonna have carbon dioxide. Okay, those two, they will bind together. They're not gonna bind, they're gonna come, and then we have this enzyme. This enzyme is very important, guys. You have to know it. Carbonic anhydrase. Okay, tamam. Carbonic anhydrase is an enzyme. It will bring like water with carbon dioxide, and it will result in the formation of this here. طبعا this you don't have to remember it. Okay, like this H2CO3, no need to remember it because aslan this compound is not stable. So once it's formed, directly it will dissociate into the hydrogen that we need, and it will form hydrogen carbonate, okay, or bicarbonate. Okay, is this clear?
Yeah, but what's the CA? The
It's
enzymes
carbonic.
that bring
Yeah,
carbonic?
it's an enzyme. It's just an enzyme that is found in the cell.
Yeah, what is it called? I just want to write it down.
Carbonic anhydrase.
Anhydrates. Okay, thank you.
Yeah, it's written here, like carbonic anhydrase. Okay. Is this step clear, guys? Okay. So
Yes, doctor.
we have our hydrogen. تمام? The hydrogen is secured. ونحن we know this hydrogen, you can see that this is, guys, you can follow the numbers. So this is number 1. It will result in the formation of hydrogen. خلاص, hydrogen is secured. We got our hydrogen. Hydrogen is secreted out. خلاص, so potassium is in. And then we have this very, like the recycling thing. طيب, hi, step 1 and 2 is done. طيب, we have hydrogen. What is missing?
The potassium, so that we can release
Chloride.
the hydrogen. Oh, chloride.
Yeah, you're correct. The potassium, but potassium is here. You can see that فات وطلع خلاص. So what is missing is the chloride. Okay. طيب, هلا chloride, it comes into the cell in 2 different pathways or in 2 different ways. So ركزوا معي. نحنا we formed hydrogen. بس كمان what did we form? We formed this, the bicarbonate, right? Yeah. So basically, what will happen is, in our body, in subhanallah, it will get rid of things it does not need, and it will bring in things that it needs. Okay. So it will kick out the hydrogen carbonate, and it will bring in the chloride. So this is the first way we get chloride. تمام? Okay, and then we have the chloride, but we need more chloride. So what happens is we have this here, this pump. Okay, this pump is called NKCC. Why is it called NKCC? Because each letter stands for the ion it brings in. It's a co-transporter. What does a co-transporter mean? Yani it brings all of the things together. A very important thing to know about co-transporters is usually the charge تبعها بيكون 0. يعني if we add the ions charge together, it's going to add up to 0. But if you can see, guys, in here, we bring in a sodium, we bring in a potassium, and we bring in 2 chlorides. Okay? تمام? Because we bring in those 2 chlorides, we're going to have a lot of chlorides. And this chloride will travel into its channel, تطلع لبرا, and that's it. That's how we have hydrogen, and that's how we have chloride, and then we result in the formation of hydrogen chloride. Is this clear? In this slide, guys, I'll give you like a tip. Just memorize the names. That's it. بس يعني إذا أنتوا بتحطوا, like if you put in your mind that what is it called? Just memorize إنه like we need hydrogen, we need chloride. How do we get the hydrogen? How do we get the chloride? And if you go through a tech in like a pathway in a cycle, it's gonna be so easy. And don't worry about it. يعني this is a very easy.
Can I
slide.
just re-explain it? البروسيس كلها عادي أنا أعيد أشرحها عشان أتأكد إنه أنا ما أكون ملخبط بشيء.
Sure.
If you don't mind.
Yeah, sure.
سوري عم باخذ من وقتكم انتم الباقية. First thing first, عندنا نحن water. والwater will CO2 they will bind together through a carbonic anhydrase to form H2CO3, which will be broken down to form a bicarbonate and hydrogen. The hydrogen will be released by the ATPase, and it will be released, but within like it will be replaced by K channel, and the K channel will release the potassium, or like the potassium, so that we can recycle it, so that we can release hydrogen. Okay, and then the bicarbonate will be replaced by the chloride through something called AE, which is anion exchanger. But we don't have carbon a lot, or like not carbon, chloride a lot. So we're gonna have NKCCl, which will release more Cl into the cell, and the Cl from the anion exchanger, and the NKCCl will be released by the cystic fibrosis TR, which will release also Cl, and they will bind together to form HCl.
تمام perfect.
سوري أخذت من وقتكم كثير.
لا عادي خالص. Moving on. تمام. This is a very like easy slide, guys. We said إنه عندنا شيء اسمه gastric. We said إنه chief cells release pepsin. Okay. The pepsin, what does it do? It's sorry, chief cells will release pepsinogen. What will the pepsinogen do? It will see the HCl. Okay. لما تلاقي ال HCl, when it sees the gastric acid, it will basically cause يعني transform into pepsin, which is the active form. Okay. This pepsin, what will it do? It will break down, as you can see here, it will break down the proteins into smaller peptide fragments. Okay. This is a very easy slide, guys, and no need to waste time on it. I'll move on. Any questions? What's the graph? Yeah, okay, the graph is, listen, it's just that, okay, the graph is that it shows pepsin works the best in acidic. Like you can see the pH in here is very low. While in here, trypsin, which is another enzyme that is not released in the stomach, it's released in the intestine. It is, what is it called? It requires alkaline. Yeah, it's just a comparison between pepsin and trypsin, which both act on peptides, okay? Perfect. And I remember in the beginning, guys, you asked me, what stimulates the release of pepsinogen from chief cells, right? Didn't you ask me this? Anyways, the answer to this is that it's the same stimulus that works on the parietal cell, okay? So whatever activates, yeah, sure, okay. I'll go over it, guys, listen. Gastric pepsin, what is it? It's basically the active form of pepsinogen. Where is pepsinogen, where does pepsinogen come from? It comes from the chief cells, okay? So the chief cells, they will release the pepsinogen. It will go up, it will meet HCl, and it will form pepsin. Tamam? Pepsin is the active form of the pepsinogen, and it will go, tamam, and it will work on the protein and break it down into smaller fragments. Now, this in here, the doctor added it just to tell you that, remember how I told you guys, HCl helps us digest protein by denaturing it. So you see, this is the protein, and it's very complicated to digest. HCl comes and it makes it easier to digest. And then the pepsin comes and breaks it down into smaller fragments. Okay? Is this clear? Perfect. Last point in this slide is, you see, guys, how we said gastrin and histamine and ACH, they bring about the release of, what is it called? The release of, what's 1 and 2? Okay, guys, 1 and 2, they're just saying, in the stomach, we have low pH, less than 4, and it's acidic, and it causes the activation of pepsin, and it causes the change of pepsinogen to pepsin, okay? I didn't go over it because I thought, خلاص, it's clear. Is it clear now?
Yes.
Yeah, tamam. So guys, just know that endopeptase is pepsin. Yeah, they're just trying to tell you. They're trying to tell you. Okay, guys, listen. Didn't we say إن إحنا عنا pepsinogen? بعدين the pepsinogen بصير pepsin. They're trying to tell you that endopeptidase, okay, endopeptidase is just an inactive form. And it requires HCl to come to form the peptidase, which is the active form, okay? Yeah. Okay, so what is it called? No, it's okay. I can, guys, listen. So basically, you all know, and we have another pulse session after the small. I might go a bit fast, and is this okay with you all?
Yeah, sure.
Yes. Come on. What is it called? I was saying, we have basically the stimulus. What's the difference between antipeptidase and pepsinogen? There's no difference. It's just a fancy name for pepsinogen. So pepsinogen is an antipeptidase. You get me? You know how in medicine we have a lot of fancy names. Yeah, it's just a very fancy name for it, and it really doesn't mean anything. Just means it needs to be activated. Okay? تمام guys, the last two sentences, and this is very important to this slide والslide اللي بعده, the chief cells, they are activated by the same stimulus that the parietal cells are activated by, and they are inhibited by the same thing that inhibits the parietal cells. يعني gastrin, histamine, and ACH, they will stimulate the release of pepsinogen, and somatostatin from these cells will inhibit the release of pepsinogen. Okay? تمام؟ خلاص perfect. Now guys, this slide is very easy. كمان نفس الشيء. Gastric acid, or sorry, not gastric acid. Here we have the gastric mucus secretion. So basically, when the gastric acid is secreted, we're secreting gastric acid, the stomach has to protect itself. So how does it protect itself? By releasing mucus. So what we have to know is, the same stimulus that causes the release of the gastric acid will cause the release of the gastric mucus. Okay? Perfect. هلا we're gonna go to what is it called? How basically gastric acid secretion is regulated. تمام? So what do we mean by regulated? هلا نحنا we're not always eating, right? So what happens basically to gastric acid when we're not eating? طب okay, نحنا we're eating. Is the gastric acid percentage always the same? Is it variable? Okay, so this is all explained in here. Okay. Let's go through the slide, the steps one by one. أول شيء, we're gonna talk about wait, I'll just check something. We're gonna talk about the vagal tone. What is the vagal tone? It's basically the tone that is kept, إنه مثلا it's the normal. The vagal tone is the usual thing that happens. When we're not eating, when we're not ingesting food, we're not doing anything, what takes over? It's the vagal tone. Okay? تمام? هلا, when we're not eating, أهم شيء guys, you have to know that the acid secretion will go from 100 to 10%. So when we're not eating, we don't need gastric acids. Like I'm not eating, there's no food in my stomach. Why would I need this very harmful thing? So what happens is that our body will try to decrease the secretion of gastric acid. Okay? Is this clear?
Yes.
Yeah, perfect. So basically, since we said we want to decrease it, does anyone have any idea how will the body decrease it? Like we mentioned something that keeps everything in check. What was it?
Somatostatin.
SST. Perfect. SST, we said it keeps everything in check. But when we're talking about what is it called? When we're talking about the basal, what is it called? The basal acid secretion. What is basal acid secretion? It means when we're not eating food, when we're not doing anything, SST is not going to be involved. Why? لأنه SST, what is it called? SST comes when it needs to prevent the release. So in here, we want to prevent the release, but we don't want to prevent it in a bad way. We want to prevent it in a way that we can sustain all day long. And مثلا some people, they fast all day long. Will their body keep on secreting SST? No. So what happens is, هون بيجي the autonomic nervous system, or in this case, the enteric nervous system. Okay? Is it clear? Did you get why SST is not involved here?
Yes, clear.
Yeah, okay. So basically, the enteric nervous system, what will it do? It's basically, what does enteric nervous system mean? يعني عنا vagus nerve. So the vagus nerve, yeah, sure. Listen, guys, we're not eating. We don't have food in our stomach. We're not even in, يعني خلاص, we're just, let's say, fasting. So our body needs to have basically a neutral, like, like a neutral position where none of the, where we don't have a lot of gastric acid secretion because we don't need the gastric acid, and at the same time we don't want to release another hormones. يعني ما بدنا, we don't want to suppress it. We don't want to release SST. So what happens is that the vagus nerve, it will take over. So what does the vagus nerve do? The vagus nerve, it will coordinate the release of HCl. How will it do that? By coordinating in other things. كيف يعني؟ يعني إحنا we said إنه to release HCl we need gastrin and we need histamine, right? So the vagus nerve is gonna send stimuli to the gastrin cells to the G cells, to release gastrin, but at a very low rate. يعني not at the maximum, يعني at the minimum. So basically 10% of gastrin will be released. So only 10% of gastrin will be released. فبعدين 10% of histamine will be released. Therefore بصير في عنا only 10% of HCl released. Did you get me, guys? يعني فهمتوا علي؟ Yeah. So بالinterdigestive phase, the vagus nerve is the big boss. Exactly. It's not regulated by SST. We don't need to suppress it. We want it to, لأنه, guys, think of it this way. SST will inhibit it. It's not going to happen. خلاص. When SST is increased, it's not going to happen. نحن in here, we want it to happen, but we want it to happen at the most minimal level, which is the 10%. Okay? So what happens? It's all Vegas's way of basically شوي. I'll tell you when does SST work. Don't worry. Exactly. Vagus is like the traffic light system. Perfect. It's like, يعني, it just allows this gastrin to release a little bit, and then histamine a little bit. So this will cause the parietal cells to secrete HCl, but what is it called? But in a very minimal amount. So between meals, it's under vagus regulation. Yep. So in interdigestive phase or the between meals, it all depends on the vagal node. Come on? Note. Okay, is this slide clear? Okay, moving on. No, we're going out of time. Hello, let's say we want to inhibit the pathway. طيب, we don't want like even the 10%, we don't want it. So what happens, guys? What comes in?
Somatostatin.
Exactly. When we don't want any gastric acid, we want for the pathway to make it خلاص not there, the somatostatin will come. So somatostatin, the D cells will release somatostatin, and somatostatin will inhibit the whole process. Hello?
Yes.
تمام. هلا in here, wait, هلا هو doctor, I'll tell you what happened. The doctor, what did he do? Why did he include those two slides? He's trying, guys, to tell you, and right now, okay, he's trying, guys, to tell you that in the between the meals, okay, we have either totally inhibition, exactly, أما total inhibition, يعني we won't have HCl at all, or we're going to have the basal or the minimum اللي هو 10%. حلو؟ يعني sometimes كمان, guys, think of it this way. نحنا, we're not eating, we need to release HCl, we need the 10%. طيب, the body released the 10%. What will happen after we release the 10%? Will we release another 10%? No, we're going to inhibit the process. You get me? So it's like it keeps it in balance. يعني, okay, we released the 10%, بس نحنا خلاص, we're finished, we don't want more. So what will we do? We release somatostatin. Okay. Hello. This is the slide, شايفين? This is the slide that explains what I'm saying. إنه نحنا in the basal secretion where it's in between meals, we're gonna have both of them. The vagal tone is gonna kick in and then somatostatin is gonna come. يعني okay, نحن vagal tone works after 10%. The 10% is out. SST is here to make it مثلا to stop the production. When the 10% is used up, vagal tone will يعني start again. You get me? It's like a balance. It's like a process. Okay? Is it clear?
بس الفيجل تون أنا ما كتير فهمته.
So listen, أنت بس just understand إنه إحنا عنا vagus nerve. حلو؟
Yes.
Vagus nerve لما إحنا نكون in between meals, what happens? It's the one that controls the secretion. So
Yes.
it will work. It will stimulate gastrin. It will stimulate gastrin, and it has a direct effect on the parietal cell. But will stimulate those two to cause the release. That's it. That's what you have to know about the vagal
بس إنه
tone.
we need like minimum. بس إنه لازم I have to know like the small steps. ولا بس إنه I have to know like the main idea and that's it.
The main idea, the overview. That's it. And guys, أنتم متى ما عرفتوا the gastrin histamine axis, all of this is easily explained to you. You get me?
Yeah, but like it stimulates. Like what does it stimulate itself? Like it stimulates the G cells,
The G cells.
right?
Yeah, the G cells.
To release gastrin, and the gastrin will do the rest of the job.
Yeah.
All right. Okay. Thank you.
You're welcome.
And then we have the inhibitory, which means the STT is involved. That's it.
Yep.
Okay. Thank you.
You're welcome. So guys, this is this slide. It it kind of says everything we talked about. Okay, and no, they're saying and no, like okay, look, I'll explain it on this like image because this image might look scary. تمام. هلا نحن we are at the 10%. تمام. So the vagus will come and then it will go and it will stimulate the G cell. حلو. The G cell will release gastrin. This gastrin will go stimulate histamine, stimulate parietal, you know the steps. Okay? Okay. هلا نحن we have an increase عنا خلص صار عنا a lot of gastric acid بيجي SST which will inhibit it. حلو؟ تمام؟ هلا there is only one thing that the doctor added here, which is that the serotonin will work here. You remember the interchromaffin cells that release serotonin? This is the step where the interchromaffin cells will come in. So what happens exactly? I'll tell you what happens. نحن we release the 10%. طب okay, how does our body know إنه نحن we release 10%? Like how would it know? So what happens is it all depends on the pH. تمام? But they see مثلا إنه oh my god, the pH صار lower than 4%. So this is like a stimulus for the serotonin cells or the interchromaffin cells to work. So they will release serotonin. This serotonin is what activates the somatostatin. Okay? Is this clear? Or did you guys get confused?
It's basically feedback inhibition. We release a lot of acetylcholine, not acetylcholine. I mean a lot of acid, and because we have a lot of acid, it stops working.
Yeah, it will cause the serotonin will be released, and then the
serotonin will be
released. Cause SST.
So,
exactly. Okay. So, guys. Yeah, sure. I can repeat. Listen. نحن, how does our body know إنه نحن we have enough gastric acid? Like, they're not gonna know إنه oh yeah we have 10%. So they will depend on the pH. If the pH goes below 4, which is the acidic, يعني the number of pH that we need, the pH that we need is 4. If the pH goes lower than 4, يعني we have a lot of acid, what happens is إنه the interchromaffin cells will get activated. Interchromaffin cells will release serotonin. Serotonin is what causes the activation of D cell, يعني the release of somatostatin. حلو؟ تمام. That's the only idea. يعني guys listen. I know there's a lot of slides. I'll show you. Wait. شايفين السلايد اللي قبله? Exactly. When it's more acidic, somatostatin is activated by the release of serotonin from interchromaffin cells. يعني if I show you the slides, look at this slide. All of these slides, they might look scary, but كله the same information. It's just that he keeps on adding many ideas, يعني many new ideas. يعني نحن كنا نعرف إنه somatostatin inhibits gastrin and inhibits everything. But in here, he just told us إنه it all depends on the pH. Well, pH will affect interchromatin cells. Okay? تمام? So I'm gonna move on because we're short on time. هلا let's say نحن we're eating. تمام? What is 5-HT? It's serotonin. It's another name for serotonin. Yeah. So basically, نحن we're eating. تمام? So I decided to eat and I'm just now chewing. Yep. So it's still in my mouth. يعني. This phase, we call it the cephalic phase. Why do we call it the cephalic phase? Because it's basically in our mouth. يعني, the food is not yet in our stomach. So when we eat, طيب, you know, basically when you eat something, guys, you're gonna smell it, you're gonna taste it, you're gonna feel it, you know, like into مثلا, let's say you're eating a burger, you're gonna smell the burger, you're gonna taste the burger, and then you're gonna start chewing. Hello, all of these, yeah, I'm doing a food ad. So all of these, basically, what is it called? They will stimulate our central nervous system. Hello. So فنحن we have food in our like mouth. This food will basically stimulate our brain. يعني خلاص يعني it makes sense. We have food in our mouth. Our brain is like, oh yeah, the food is coming. So what does it do? هون it will act on the vagus nerve. It will increase its activation. إنه يلا get activated. It will increase its activation up to 30%. Okay? So increasing the vagal activation means we're gonna increase gastrin. Gastrin will increase histamine. Histamine will increase شو اسمه HCl. You get me, guys? فيعني لما يكون في food in our mouth, this will favor increased vagal activation. يعني we're gonna have more and more release of the different things. Okay? Okay. هلا in here.
So the acetylcholine basically in here in the vagus tone, it acts, it activates the epithelial cells.
Yeah. It activates gastrin.
Gastrin.
تمام؟ يلا. Moving on. هلا the food is in our esophagus. It's no longer in our mouth. The food is in our esophagus and it's about to enter the stomach. Okay? So we have certain factors that will increase the acid secretion. يعني if you guys can see, wait, I'll show you. In here, the acid secretion was 30% and the food was still in our mouth. But in here, the acid secretion is 60%. But why is the acid secretion 60%? So what happens is, once the food enters our stomach, it goes through the esophagus all the way down to the stomach. It will cause the stomach. Remember, guys, oh no, wait, I'm gonna explain this in the next lecture. But when we eat, our stomach will extend, right? It will distend, it balloons up with the food, right? So since it balloons up or it extends, what happens is this extension, it will activate the intrachromatin cells, because intrachromatin cells, they have something called mechanoreceptors. Mechanoreceptors are receptors that basically they sense any change in movement, they sense the stretch, they sense the hey, okay. So this distension caused the activation of what is it called? The activation of intrachromatin cells. فبصير عندنا release of serotonin. حلو? Now, what will serotonin do? Serotonin in here, okay, it will do the opposite than what it did يعني before. In here, بالعكس, it will cause the vagal reflex to be activated. Okay, so when we activate the vagal reflex, what happens is the vagus nerve will start firing like crazy. فخلص vagus nerve will start firing more and more and more and more. وبصير في عندنا increased what is it called? Gastric secretion, increased histamine, increased HCl. Okay? Is this point clear? Okay. And we said نحنا إنه serotonin is like, it's like a, what is it called? It's like a hormone that acts on stimulatory and inhibitory neurons. فهلا in this case, serotonin will inhibit these cells. It will inhibit somatostatin. Why? لأنه نحن we need the vagus to work more than somatostatin. Somatostatin we want to like خلاص inhibit it. We don't want any inhibition. We want our maximal for like acid formation. Okay? So this is the first, this is like the first stimulus. تمام؟ هلا we have two more stimuli. What are they? The second stimuli are the presence of peptides. So نحن when we eat, our food is composed of many different things, carbs, peptides, whatever. So basically the presence of peptides, they're going to basically, what is it called? They're going to be detected by the G cells. But the G cells, when they see peptides, they're like, oh no, we need to increase our like work. But they will start increasing their work. I'm still going on through it. Wait. Yep, exactly. Serotonin will get inhibited when acidity increases. Wait, this is the point I didn't go over yet. Wait. So guys, someone is asking me to repeat the slide. Okay, هلا أنا لسه ما خلصت the slide. I only finished the first step, so I repeat the first step. نحنا when the food reaches our stomach, okay, we need to increase the formation of HCl or acid secretion. This is increased by three different things. The first thing, it's basically the distension of the stomach. يعني أنت your stomach was empty and suddenly it ballooned up with all of these foods. Okay, so what happens? What happens is that you see here those serotonin cells. They are what is it called? They have mechanoreceptors. What does mechanoreceptor mean? A mechanoreceptor is a receptor that يعني إنه خلاص when emotion happens, when something extends or like compresses, it's gonna detect this change and it will release serotonin. طيب, as we said, serotonin, it acts on stimulatory and inhibitory neurons. Okay, so serotonin will go and it will bind to the stimulatory, guys, stimulatory, and it will stimulate stimulatory neuron لعند الفيجاس, and it will increase the vagus release. So the vagus will start firing like crazy. Okay? Tamam, is this obvious? Is this clear? Guys? Okay. Perfect. So you guys think this is clear. With this thing, yeah, this thing is called vagovagal reflex. Vagovagal reflex is what happens in here. The vagus is activated by the serotonin that is released by the intercolumn cells because of the distension. Okay? Tamam. Hello. The next thing that increases the gastric secretion is the presence of peptides. You can see it here. So basically, نحن when we eat, you know, like food is made up of carbs, of proteins, and so on. So نحن we have peptides, okay? Those peptides, they will, what is it called? Those peptides, they will go, يعني they will be detected by G cells. When G cells sees peptides, it's like, okay, خلاص, I have food. يعني, you know, like G cells are like, oh my God, we have food. It's actually there. But they start releasing gastrin, لحالا, like they start releasing gastrin, okay? Is this clear? Guys, so this is clear. Last but not least, the last thing that affects this is the pH. The doctor called it food buffer. So basically, we ate, so we have a lot of food. The food will mix with the acid. So zaman, when there was no food, يعني we only think the acid concentration was the acid itself. But when food is there, it will combine with the acid, so the acid will be diluted. So this will buffer, the food buffers the acid, as we can see here. So the pH is going to go higher. It's not going to go like from 4 and below. لا, it's going to go towards an alkaline. Hello? Is this clear? So when the pH increases and it becomes more alkaline, this will inhibit those interchromaffin cells from releasing serotonin Therefore, it will inhibit the activation of SST. Is this clear? تمام. So guys, بس عرفوا إنه interconnecting cells in here, they're affected by pH. If it's more acidic, they're activated. If it's more alkaline, they're deactivated. And SST depends on that. Okay? Is this slide clear? Let's see what's the next. Is this slide clear? The last part, yeah, sure. So basically, when we eat food, the food is going to mix with the gastric acid. So when it mixes with the gastric acid, the pH of the stomach is not gonna, like, relax. The pH is gonna become more alkaline because we're kind of neutralizing the acid with the food contents that we have. Okay, for those food contents, what will happen is, the pH is gonna rise. It's gonna be more than 4. Now, when the pH rises and it's more than 4, this will inhibit the interconnecting cells from releasing serotonin. فهلا هدول interconnecting cells اللي هون، okay؟ يعني شوفوا هدول، يعني guys, those are 2 different cells in 2 different positions. هدول they're usually in the fundus. هدول they're more in the pylorus, okay? The pyloric region. So basically لما ال pH بيقل، بالعكس ال pH بيعلى، يعني pH is greater than 4, they will get خلاص. They're gonna get inhibited. So because they're inhibited, SST is inhibited. Okay, so we're gonna favor the release of gastrin. تمام? So عندنا more acid secretion. Perfect. In here, this shows how everything goes back to normal. تمام? يعني if you think of it, guys, نحنا we cause distension, we cause decreased pH, we cause peptides. We have peptides. If we remove all those 3 stimuli, what will happen? خلاص, the vagal nerve will not be stimulated. The interconnecting cells will not be stimulated. Well, pH is gonna go back to normal. It's gonna go back to become more acidic. فلأنه it becomes more acidic, the interconnecting cells will activate the somatostatin. بصير في عنا inhibition of this process. Okay? And just think, everything I just explained in the other slide is gone, but it goes back to normal. فبنرجع للسوماتوستاتين. حلو? Should I move on? Yeah, sure. Listen, in this slide, we're just saying, خلاص, the food that was in our stomach left. It's now in the duodenum. It's now in the small intestine. So our stomach is back to normal. خلاص, it's no longer distended. We don't have peptides anymore. And the pH is going back to below 4. So it's becoming more acidic. And احنا حكينا لما pH goes back to below 4, what is stimulated? Interconnecting cells. So لأنه pH is lower than 4, يعني it's acidic, it's gonna be stimulated. تمام? But because they're stimulated وعنا acidity is increased again, بصير في عنا interconnecting cells, they release serotonin. This serotonin will cause the release of somatostatin. Somatostatin is gonna inhibit gastrin production because خلاص, we don't need gastric acid anymore. Okay?
It's basically everything what we said before. It's just that food is in duodenum, that's it.
Yeah,
so it became...
exactly. We have this slide, guys, is the last slide in gastric secretion. I'm gonna explain this and you can guys go have a break. I'll give you a break for like 5-10 minutes. And we move on to the emptying and filling, okay? Is this good? Come on. Okay. So basically, this is the last thing. So we said إنه الfood راح من our stomach all the way into the intestine, okay? So what happens is إنه the intestine كمان, it affects the gastric secretion. And I know this sounds crazy, but it's true. So what happens is, and we said the food went from here, from the pylorus, all the way down into the intestine. So when it goes there, the fatty acids, okay, they're going to release something called, the fatty acids, not themselves, they're going to release. So the fatty acids رح يجوا, and they're going to meet something called I cell. The eye cell is usually found in the duodenum, okay? This eye cell, once it detects the fatty acid, it will release something called CCK, okay? So CCK is released, and then didn't we say, no, basically the acid contents are basically acidic. So the duodenum is not used to have something acidic. So when we have acid, come on, it will stimulate a cell called S cell, and S cell will release secretin. So now we have CCK and secretin. Those two, they will go and they will activate the vagal nerve, okay? They will activate the vagal nerve, but in a bad way. And they will activate it in an inhibitory type of fashion. So the vagus nerve will get activated. It will go لعند somatostatin, okay? And it will tell the somatostatin, يلا, no, release, sorry, it will go لعند الدي cell, and it will be like, يلا, release somatostatin. So by then we start releasing somatostatin, which will further decrease the gastric acid. Is this clear? Yeah, just know, guys. Listen, the one you're talking about is a receptor. This is not a receptor. This is a hormone. This, you guys, yeah, don't confuse this CCK with the receptor, the gastrin receptor. This CCK is a hormone in here. It's not a receptor. In the parietal cell, CCK2 is a receptor. In here, it's a hormone that is released by duodenum. I know it's the same name, but يعني in heck they put it, okay? So what happens is, the fatty, I'll give you a review of this. The fatty acids and the acidic, what is it called? The acidic environment of the stomach, of the chyme, yani, that is released from the stomach, it will stimulate eye cells and it will stimulate S cells. But eye cells and S cells, they will release CCK by eye cells, S will release secretin. Those two will stimulate the vagal pathway, the vagus nerve, to activate the D cells so that somatostatin is released. Okay? تمام? Guys, this is a very easy slide. خلاص يعني. تمام. Thank you. So basically, guys, in here, this is you can guys read it alone. It's like a summary of everything, and I'm not gonna go over it صراحة. I know we explained everything a lot. You guys have another, like we have now this, the filling and emptying. Wait, so eye cells secrete CCK, which stimulate S cells, which release secretin. No, eye cells, wait, let's go back. Eye cells, they will release CCK. They are stimulated by fatty acids. S cells are stimulated by the acidic composition of the food. So the food that we eat, يعني الفود اللي طلع من السtomach مو راح على gastric acid, it's strained with the acid and stuff. You get me? But because it's coated with the acid of the stomach, it's gonna stimulate basically the release of secretin by S cells. You get me? So they're two different processes. Is it clear? Yeah, okay. So guys, as I said, those two are in division. You can go over them, but I'm sure, no, guys, you're gonna know it. Hello. Extrinsic or intrinsic. Do you know what does intrinsic and extrinsic mean? Yeah. Exactly. Both of them, don't listen, guys, لا تضيعوا حالكم بالdetails. Just know the overview, لأنه there's a lot of details that are not important. Just know CCK and secretin, both of them. Yeah, sure. That's the plan. I'm gonna give you 10 minutes of break, go pray, and then we're gonna start the next one. So what is it called? CCK, secretin, they're just gonna act on the vagus nerve, and the vagus nerve is gonna cause what is it called? The D cells to secrete somatostatin. Well, pause for good. Perfect. So guys, هلا you can go have your break. Inshallah by 5:12 we're gonna complete. تمام? You're welcome. Anyways, shall I start? يلا. Guys, can you hear me? خلاص بلش? Okay, perfect. So now we're going to discuss the gastric filling and emptying. It's way simpler than the gastric secretion. And as I mentioned earlier, guys, if you have any questions, if you have any doubts about gastric secretion and about this, if I couldn't answer you in the pulse session itself because we're short on time, here are my details. This is my email. Oh, they sent an email saying it's at 8. Okay, perfect. Then you have enough time. Inshallah, yani, it's not gonna take so much. It's way easier than before. And here's my number, guys, if you need anything. Don't be shy. حتى if you want me to explain to you anything in uni face to face, I'm more than happy to do that. Just text me and I'll be there, inshallah. Now let's start. Tamam. Wait. Okay. So now we talked about gastric, like the what is it called? The stomach from the point of view of mucus, from the secretions and stuff like that. In here, we're gonna talk about it in the point of view of muscles and contractions and stuff like that. Okay? If you think about it, what are the main three functions that the stomach has to do? Does anyone have any idea? Yani? Just tell me anything that comes to your mind. Store food, exactly. That's a very good one. Chemical and mechanical digestion, perfect. Mix, perfect. All of the questions, gastric secretion, exactly. All of the questions you gave me, guys, are like amazing. We're gonna summarize them in 3 steps, which is first of all, accommodation. What does accommodation do? Or mudu, I mean, what does accommodation mean? Accommodation means, it's going to make space for the food. Because when we eat food, if you guys think of it, our stomach is not as big as we think it is. It's probably like if you guys went to the lab and you saw it, it's so tiny. It's like what? It's like this, Like, so if you think of it, the amount of food we eat has to all be stored in our stomach. And what is even more crazy is the idea that it's not stored in all of the stomach. There is a specific area for storage and for accommodation before like actually starting the contractile activity. So in what is it called? Guys, in short, I mean, accommodation is all about letting food in. Okay, so accommodation is a very important like function of the fundus. Okay, if you can look at the stomach in here, the fundus, هيك شايفين how it's very like smooth, and you look at here how it's wrinkly and ugly, but in here it's smooth. Can you see it? So basically, the idea is in the fundus, it is the place that is responsible for accommodation, because in the fundus we don't have peristaltic movement. Then we don't have peristaltic movement, it's all like smooth, it's not wrinkly and stuff. So yes, our stomach has to accommodate our food, and it doesn't only have to accommodate it, it has to accommodate it with no pressure changes. What does no pressure change mean? You know how مثلا in our chest area the pressure in our lungs is at a specific degree, and if the pressure increases or decreases, this might cause مثلا any pathology or any disease to happen. The same thing happens in our stomach, by the way. So basically, when we take in food, we don't want the pressure of the gastric content to increase very, very rapidly. This will cause basically herniation. It can cause many different problems. So subhanallah, God created us in a way where basically our stomach accommodates the food without causing any change in pressure. It's like nothing happened. Okay, this is the first function. The second function is that it has to do peristalsis to basically mix and churn the food to make it into smaller and smaller particles, and you can see it here. Here is the peristalsis. You can see the wrinkly muscles here, and you can see how those like particles were way larger and they become like smaller and smaller. Okay, lastly, it has to empty all of this into the duodenum, and it cannot empty it into the duodenum unless it's in a specific form. It has to be in the chyme form, and it has to be small particles. And you can clearly see, guys, these particles in here are way different than the particles in here, right? Yeah. Basically, to do that, the stomach has to keep on contracting and relaxing, and has to have peristaltic movements and so on. But now we're gonna go like in details and talk about every single function. Okay. Remember how in the stomach, in the previous lecture, we said the secretion, the gastric secretion had its own functional divisions. Well, in here, come on, gastric motility has its own functional division. The functional divisions are proximal and distal. You can see, guys, in here, the proximal is the fundus. You remember how I told you guys the fundus is there to accommodate? So here, the fundus is here to accommodate. And then we have the distal stomach, which is responsible for, responsible for gastric mixing. And you can see here, guys, it's all wrinkly and peristaltic and so on. Is this clear? It's pretty easy. There's nothing much to talk about here. Okay. And yeah, we're gonna talk about the proximal stomach. Okay, we're not gonna go into the distal, we're only gonna talk about the proximal stomach. The proximal stomach, as I mentioned, it's made up of the fundus. What does the fundus do? It is there to accommodate the food. How does it accommodate the food? It accommodates the food by show, guys, by tonic, this is very important, tonic relaxation and tonic contraction. Okay, this is very, very important. It's okay by stretching. Stretching is another, it's the easy word, but technically it's a tonic relaxation. Why is it important that I'm telling you guys it's tonic? The word tonic means that it will maintain its tone. We're not gonna have any slow waves. You can see, guys, in here the doctor said no. Sorry, right, moving the chat. So you can see, guys, and no, the doctor in here said that we have no slow waves. What are the slow waves? They're basically the peristaltic waves. So in here, in the fundus, in the most proximal area of the stomach, there will be no slow waves. There will be no contractions, like no peristaltic contractions. Okay, this is very important. What will be there, on the other hand, tonic and tonic relaxation. Okay, so when we eat, you know that our stomach can hold up to 1.5 liter of foods without having any changes in pressure. So where does all of this food go? It goes here. So the food is in here. What happens is, look, I'll show you. Okay, what happens is when the food is going all the way down from your mouth into your esophagus, there is something. You remember how Ghaid yesterday told you guys, when you eat, the food is going to go into your pharynx, and then your pharynx will stimulate basically the lower esophageal sphincter to relax, so it allows the food to pass by. Do you remember this? Yes, perfect. So basically, this action on the lower esophageal sphincter is the same action on the stomach. So the pharynx, once food hits the pharynx, it will activate the what is it called? The vagal nerve. فبصير في عندنا vagal reflex, and this vagal reflex بصير عندنا in here the pharynx is full. It's not here, but guys imagine it. So it will send signals all the way down here to the what is it called? To this, to relax. طبعا relaxation is what is part of an inhibitory type of movement. So basically, the vagus nerve will send acetylcholine to the inhibitory neuron. The inhibitory neuron will release what I think I said, covered it yesterday. It will release nitric oxide and VIP. Nitric oxide and VIP are two important hormones to bring about what? To bring about relaxation. Okay? Is this clear? So basically, where the what is it called? The fundus will relax, and it's not only this is just, guys, the same thing. It's called receptive relaxation, because the pharynx in here will send will send what is it called, through the vagus nerve, will send what is it called, will send like signals into the, what's the difference between tonic and peristaltic contraction? Okay, that's a very excellent question. Tonic means, you know, guys, how do you remember sphincters? Do you know the sphincters? Sphincters have a tone. What does a tone mean? It means that it maintains its shape, it maintains its height, but it does not contract, you know, by like a unit. As a tone, tone, like I'll show you. Wait. You see here, the whole fundus will contract or relax together. It's not gonna be like peristalsis, exactly like rubber band. It's not gonna have waves. It's gonna خلاص. They will all contract together. They will all relax together. Okay. While in here, in the peristaltic wave, it's not gonna be like that. In a peristaltic wave, you see, مثلا, in here we have contraction, but in here we have relaxation. So this band in here, like this part of the like stomach, is not going the same, it's not undergoing the same process as the part in here which is contracting. Did you get me? So it's not acting all together. Relax. Everyone is at a phase. One is relaxed, one is contracted, and so on. Yeah, that's right. تمام. هلا في كان a question what was it? No, wait. I know what does it wait? This is stimulated by what? Okay. What I was saying is, the pharynx, when we have food, okay? Yeah, exactly. When we have the food, we ate something and then the food hits the pharynx will send signals via the vagus nerve, and no, we have food. The vagus nerve will go all the way down. تمام. It will release acetylcholine that will bind to basically muscarinic cholinergic receptors. Those muscarinic cholinergic receptors, they will activate inhibitory neurons. Okay, this is important. فلأنه they're inhibitory neurons, what are they going to send? They're going to send, or secrete, they're going to secrete VIP and nitric oxide. Okay, so when they secrete the VIP and nitric oxide, we're going to have the receptive relaxation. Is it clear? Tamam. Now, not only the pharynx activates the vagus nerve, كمان our own stomach, okay? Our own stomach, when the food reaches the stomach and it's there, the what is it called? The stomach itself كمان will activate the vagus nerve, which كمان will activate an inhibitory neuron, resulting in the release of VIP and nitric oxide. So we have two things that are causing the release of VIP and nitric oxide: the vagovagal reflex by the pharynx, and this is called receptive relaxation, and the vagovagal reflex by the stomach, and this is called accommodation. Okay? Is this clear? Both of them are the same pathway, but the only different thing is in the receptive relaxation, it's initiated by the pharynx. In accommodation, it's initiated by the stomach. Okay? تمام?
So basically, when the muscle is contracted, and there's a lot of food, we like the stomach. It has receptors, so it will stimulate it will stimulate the inhibitory neurons to release VIP and norepinephrine, or not norepinephrine, NO.
Nitric oxide.
So, yeah. So these two neurotransmitters or hormones will make it relax, right?
Yep, it will cause relaxation.
Right. So we have receptors we have like mechanoreceptors in the stomach that detects the pressure.
I'm gonna talk about this in a bit. Just for you guys know, and once the food is there, the food being in the stomach will cause the release of
And in the pharynx, for the LES اللي هي lower esophageal spinator. Okay, so we have LES وبعدين عن accommodation اللي هي in here.
هلا مو ال LES, the other one is called receptive relaxation. I told you guys إنه whatever happens to the LES happens to the stomach too. Okay, يعني just to correlate, تمام. So it's stimulated by gastric distension. You can say that, but I think we're gonna like go over exactly what happens in a bit. Yeah, here. So what happens is, guys, as we said, إنه distension of the, what is it called, the stomach will activate the mechanoreceptors, which will eventually activate the vagovagal reflex. تمام? Is this good? The only added information in here is that, remember guys, حتى gastric secretion, what cells where contains mechanoreceptors? يلا guys, مثلا دقيقتين talking about it. What cells contain mechanoreceptors inside the stomach? EC cells. Perfect. Intercalatin cells. Yep. So basically, the EC cells, they will release, and in this slide, you can see, guys, that the distension or the ballooning up of the stomach will cause the intercalatin cells to release serotonin, and this serotonin will what is it called? It will act on like the neurons that are close to it. يعني شوفوا مثلا, this is what he means by local enteric nervous system reflex. So basically, the EC cells, they release serotonin that will act on muscarinic receptors. Yeah, they will release. Sorry, serotonin is released. حلو. It will go to this neuron. This neuron is a part of the enteric nervous system. This neuron is activated, so it will release acetylcholine. Acetylcholine will then cause the release of, basically will stimulate the release of nitric oxide and VIP by an inhibitory neuron. Okay? Is this clear? It's like a steps, يعني. It's the same step on and on and on. Once the ECL is distended, serotonin, ECH, and so on. حتى guys, you can see in here that this is the vagovagal reflex. عندنا هون كمان الtension can activate the vagus nerve. Okay, the distension will activate the vagus nerve, which will كمان act on the inhibitory cells. So the distension, mechanoreceptors sense tension and stimulate vagus nerve to release ACH, which will stimulate the inhibitory neuron to release VIP and nerve, which will relax. Yep, that's exactly what happens. And come on, adding to what your colleague just said, where there's serotonin in it. Wait. Mechanoreceptors stimulate. Basically, the mechanoreceptors are the EC, so they will secrete serotonin. Serotonin is going to bind to the vagus nerve, which will get it stimulated. You get me? So how is vagus nerve stimulated? By the serotonin that is released due to basically the distension. And guys, we have local enteric nervous system. So basically, بدل ما نروح على vagus nerve وهذا الطريق الطويل, like this long pathway, we have enteric nervous system in here. It can just directly, serotonin can go and activate it. So it's activated by both the vagovagal reflex and the enteric that is just there. Okay? Is it clear?
So we have two pathways of inhibition. It's either by extra EC cell, which releases serotonin, which activates the neurons, and the neurons will release an acetylcholine, and the acetylcholine will release NO-VIP, or what's the other pathway?
The vagus nerve. Vago. And technically it's the same thing. It's just that the serotonin is released. It can either go to the vagus nerve or the local enteric nervous system cells. You get me?
So the vagus nerve are gonna release the NO-VIP, right?
The ENS. Okay, guys, look. No, look, guys, ركزوا معي. We have.
I got confused.
Yeah, sure. It's okay. We have the stomach balloons up and it distends. So yeah, I'll like point in here so you can guys see. Tension, we have it. The tension is released because of the distension, and this will stimulate the intraconfident cells to release serotonin. Okay, so this serotonin, it will go to this. شايفين هاي? Can you see this, guys?
Yes.
This neuron is an enteric nervous system neuron. So it's a neuron that is specific for the stomach. تمام? So basically, what will it do? It will go stimulate this neuron that will release acetylcholine. This acetylcholine, when حيروح على this neuron, what is this neuron? It's an inhibitory neuron. This inhibitory neuron is what will secrete nitric oxide, GIT. And the same serotonin can take another pathway, which is this vagus nerve. Hello, you guys have to know, and the vagus nerve is like the biggest nerve ever. يعني خلاص, it can moderate a lot of different autonomic activities. That's why it can command serotonin can go to the vagus nerve and then stimulate. The vagus nerve will go, it will go. This is the afferent and this is the efferent of the vagus, and then it will go to the inhibitory right away, release acetylcholine and activate it. Okay, is this clear?
Yes.
No, there is serotonin in both ways. Relax. Think of it, serotonin is what starts all of this. You see, the serotonin is what activates the ENS. And it activates the vagus. It activates both of them. The only difference is that ENS, if you think of it, there's literally no difference. But it's just that the position, the ENS is locally found in the gastric. The vagus, they have to go all the way back to the brain. And then, but you see, guys, look, ENS is just one. It's just this neuron. But for the vagus, we have the afferent and then we have the efferent. So it has to go to the brain and then to the, and it's just a longer pathway. But in here, ENS, it's the shorter pathway. Is it clear? It's literally the same thing, guys. Serotonin.
So afferent means it goes to the D. بس إيش المعنى الافferent والefferent? I forgot.
Afferent is just that it takes the stimulus from the stomach to the brain. Efferent from the brain to the stomach. Okay.
Okay, so we have two, like short pathway and long pathway. That's it.
Yeah, you can think of it that way. The short pathway is the ENS. The long pathway is the vagovagal reflex, and both pathways are stimulated. Is it
okay?
All right now, guys?
Yes.
Do you have any questions? If you want me to repeat, I don't mind repeating. You guys tell me. And if there's something that is confusing you, come on, tell me. Come on. Plus, I'll move on.
So kind of it's the nervous system that releases acetylcholine.
What?
We have neurons that get activated from the sternoton, I guess, whatever you want to call it, and it releases acetylcholine so that it activates the inhibitory cells.
Yeah,
both of
them. Both of them will release acetylcholine. The vagus nerve and the ENS nerve. Both of them will release acetylcholine.
Okay, it's called it's called NES nerve.
Yeah, here, what does ENS mean? It means enteric nervous system. Enteric nervous. Yeah, what enteric means, everything that is related to your GI is enteric, but it's just a special name, okay? It's the same thing, huh? Hello. After we ingest it and it is stored and everything, what happens is that we need to empty those those particles. We don't need them anymore in the fundus. We need to start digesting them and so on. So what happens is we have something called fundic emptying. What is fundic emptying? Fundic emptying is when basically the fundus contracts. بصير في عنا tonic contraction. This tonic contraction بصير هيك whoop, but we'll just push the food down. تمام? Is this clear? It's pretty easy, guys. خلاص, contraction, emptying, next. تمام. Perfect. Now we're gonna go into the distal stomach. The distal stomach in here, it's different than the fundus. Why? Because, guys, as you can see, هون بخلص الفندس, and then we have this very unique area called the pacemaker region. What is the pacemaker region? The pacemaker region is the region that releases it releases the basal electrical rhythm. يعني it decides the basal electrical rhythm. What is the basal electrical rhythm? It's basically electrical signals that stimulate the peristaltic contractions. Okay, so the pacemaker region is like the big boss in here. It determines, okay, I want to release 3 waves per minute. So it will cause basically 3 signals to be released every minute, and those 3 signals will bring about the peristaltic contractions. Okay, is this clear? Is it? Let's move on. Yeah, sure. So basically, guys, we have this thing here. It's called the pacemaker region. The pacemaker region, it's like the big boss of the peristaltic waves. شو يعني? يعني نحن we know that basically in this area, the distal stomach, we have a lot of peristaltic waves. But what is controlling those peristaltic waves? Are they just take من عقلهم, they're working by themselves? No. We have this pacemaker region will release electrical signals in a certain rhythm. This rhythm is called basal electrical rhythm. Okay. So they will release the what is it called? The electrical signals in this rhythm. This rhythm will dictate the muscles, and okay, now you have to have 3 peristaltic waves per minute. Okay, so they are what, يعني basically this region, the pacemaker region, is what determines or dictates the amount of contractions that we need to have. Okay. Is it clear? Okay. So, guys, in this region, we have the breaking down of the particles from big particles into smaller particles, and we have basically the, what is it called? The like digestion. You remember that we release pepsin, we release lipase, and so on. هلا pepsin, I forgot to mention, إنه pepsin and lipase in the stomach. Yeah, sure, I can repeat, don't worry. So guys, listen. Again, we have this region called the pacemaker region. This blue zone, okay? You can think of it as the cells that control all of the peristaltic waves. But how do they control the peristaltic waves? We know for any contraction or any relaxation, we need an electrical signal, right? Yes. So basically, the pacemaker region, it will release electrical signals at a certain rhythm. And this rhythm is called the basal electrical rhythm. So the basal electrical rhythm is what dictates, or like it tells the muscles that, okay, now guys, you have to have 3 contractions every minute. Okay. So what happens is, it will release electrical signals. Those electrical signals will be detected by the muscles, and the muscles will start contracting, relaxing, contracting, relaxing in a peristaltic type of way. Okay, is it clear now?
Yeah, so basically in the pacemaker region, it's like it acts, it's like a brain. It's like cells that it tells that we should like it tells the cells, like these cells, these specific cells, they stimulate they stimulate the other muscles to make 3 waves. That's it. In a rhythm. In a rhythm called BER.
Yeah, and you can think of it that way, true. Yeah. تمام. هلا moving on, guys. What happens is that as we go further away from the pacemaker region, the strength of the waves gets stronger and stronger. Can you guys tell me, why do you think the strength gets stronger and stronger? Do you have any idea why? To contract food more, to make chyme. Yeah, exactly. And to push the substances. Both of them are correct. So what happens is, we want to break down the food. We want, what is it called? We want smaller and smaller particles. So to basically achieve that, what happens is, in this slide, you can see, guys, the doctors placed sensors, okay, in different areas of the stomach. يعني هون it's in the fundus place. Here's the pacemaker region, and as we go down, it's going more towards the pyloric region. Okay, and then they calculated two things: the electrical waves. The slow waves are basically electrical waves, and then they checked the contraction and the strength of the contraction. So if you guys compare, and let's go step by step. First one, in here in the fundus, we said نحن إنه we have no slow waves, we have no peristaltic contractions. We only have tonic contractions, right? Right. That's why we can see no waves in here. But then when we go to the pacemaker region, we can see that, oh yeah, there is an electrical wave in here. And this electrical wave is bringing or causing a contraction and then a contraction. تمام؟ هلا if we go لتحت أكتر يعني if you go down and we compare them, you can see that the electrical signal is going like هيك بتصير more rapid. You see how there's a lot of like هيك small attenuations, and if you look in here, you look at the muscle, like the contraction and the amplitude, you see how it increases. يعني compare this, you see how tiny this is, and then compare it to this. But they realized إنه نحن as we go further away from the pacemaker region, as we go closer to the pyloric region, the strength of the waves, the strength of the peristaltic waves is going to increase more and more and more and more. Okay, the last one, this one, yeah, number 5, or like the point. I'll repeat it all. So basically, guys, as we said, إنه نحن عندنا sensories in different areas. We said بالfundus we don't have any peristaltic waves, so we don't have لا electrical stimulation ولا حتى muscle contraction. But in the pacemaker region, we're going to start having the electrical waves. طيب, this is the first electrical wave. It will cause this tiny contraction. And then as we go down and down and down, you can see, guys, how basically the electrical signal itself is getting more, and you can see more spikes, more spikes, more spikes. So basically you see here we have only 2 spikes, and here we have like 3 spikes, and here we have 5, 4. So it's keep on increasing. And this increase in the spikes and in the electrical signal will cause an increase in the amplitude of the muscle contraction. You can see that the amplitude of this muscle contraction compared to this muscle contraction is way stronger, صح? It's just that
sorry قطعتك, but in other words, as we go down like the stomach, or like as we are going to the duodenum, basically, the waves of the electrical waves, we're gonna have it like stronger, which means we're gonna have more muscle contraction, or like stronger muscle contraction as we are moving down the stomach.
Yeah.
غلط?
No, صح.
Okay. And more spikes, يعني.
Yeah, more spikes more spikes means we have increased amplitude and increased strength. Okay, is this clear, guys? Hi guys, is this clear?
Yeah,
I'm
yeah.
thinking for you. Like, هلا as we said, I asked you guys in why do we need the strength of the, like the, what is it called, the strength of the contractions to increase? Here we explain why. Look, so first of all, we have 3 contractile waves per minute. This will cause, when we have a يعني هيك شايفين في عندنا contraction, the antrum contracts. This contraction will force the pylorus to open. Okay, now the pylorus is open. What will happen? Those small tiny particles will go in, اللي هو basically the chyme. Okay, but those large particles, they're not able to pass in here because they're larger than the pyloric opening. Pyloric opening only allows particles that are less than 2 millimeters to pass. So what happens is, as they try to move, they're pushed backwards. وبصير في عنا مثل a contraction backwards that is so strong. Okay, this contraction backward will force the pylorus. It will force the pylorus and the antrum shut. يعني شايفين كيف in here the antrum was contracting, pushing these in here, and the pylorus was open, and it allowed the small particles to pass. But once the small particles all passed and the large particles are trying to move in, yeah, sure. So first of all, the antrum is going to contract. This contraction in here is going to push, and you can see all these particles are pushed against the pylorus. So the pylorus will open, and the pylorus will open, and it will cause the small particles that are basically less than 2 millimeters to go there, to pass. Khalas, they passed, you see? Those passed, and they went into the duodenum, okay? But when the large particles start to pass, yani the large particle is trying to pass, they can't pass through the pylorus. For the body, what will it do? To prevent basically another particle to obstruct the pylorus, the stomach will contract very strongly, but backward. Okay, يعني it will contract in here. You see this area here, it will contract to push those particles backward, and those particles will go all the way back. The pylorus will close and the antrum will close. But why does this happen? This happens to force those large particles back into the stomach so they can get crushed more and more to form the small particles. Is this clear? Do you want me guys to repeat? It's clear. Okay, perfect. If anyone wants me to repeat, Adi. Okay, so and this, guys, this forceful contraction in the opposite direction, and it's not pushing the food there, it's pushing it back, is called retropulsion. Hello? Retropulsion is a really important word. You guys have to remember it, and you have to know. And retropulsion is important for large particles to get back into the stomach and get again more grinded, more mixed, more broken down, and so on. Okay? Okay. Now, moving on. Remember, guys, how did we have like cephalic phase, gastric phase, intestinal phase, gastric secretion? We have the same thing in here. In our stomach, okay, if we're not eating, it's not going to contract. Like, why would our stomach contract if we're not eating? So what happens? What happens is our stomach goes through this phase called the migrating motility complex. Okay, this is a very important complex that usually happens between meals. Tamam? So what is this complex about? So let's say I'm not eating, my stomach is relaxing. Okay, my stomach does not have any food, I'm not eating, I'm not gonna eat anytime soon. So what will the muscles do? Will they contract? Will they relax? So this thing, the migrating motility complex, or MMC, you can guys think of it as a cycle. Okay, it's a cycle that is made up of 3 phases that I'm going to explain now. And these 3 phases, or this cycle, will keep on repeating every 90 to 120 minutes. And every 90 to 120 minutes, the phases will be completed, the cycle will be completed. If there is no food, then the cycle will repeat itself again. But if there is food, then the things that we talked about will happen. Okay, is this clear? Is that? So basically in phase 1, so I told you, it's made up of 3 phases. So what is phase 1? Phase 1 is the phase of relaxation, or the phase where we have nothing, like zero, no contraction, no tonic contraction, no peristaltic contraction, nothing. Okay. And this phase is basically, it makes up 50% of the time. يعني مثلا 90 minutes, 50% of it is like 45 minutes. So we have 45 minutes of relaxation. The stomach is not doing anything, there's no contraction. Okay? Phase 1 is followed by an erratic, non-propulsive phase, which is made up of basically rhythmic contractions. So this makes up 40% of the time. But 40% of the remaining time, the stomach will start having rhythmic contractions. Okay, and it's just contracting like this. It's not doing anything. The contraction is not there for any reason, but it's contracting. Okay, and then this contraction will suddenly become very intense. Peristaltic waves, very intense peristaltic waves, and this is basically phase 3. And it only lasts for 5 to 10 minutes. Okay? Is it clear? So basically phase 1, there is no contraction. There is nothing. Everything is relaxing. Phase 2 بيصير عنا rhythmic contractions. يعني هيك they contract slowly all together. There is no peristalsis. Only all the stomach will contract. And then we will go, this will like prepare the stomach for phase 3, which is very short. It's only 5 to 10 minutes, where we're going to have very intense peristaltic waves. Okay? Hala, it's really important, guys, to remember that this complex or this cycle is stimulated by what is it called? It is stimulated by motility, which is an enzyme, not an enzyme, it's a hormone that is secreted by the duodenum. Okay? So motility, I think it's very easy to memorize this. Motility, يعني احنا we're talking about the gastric motility. So motility is released to basically, what is it called? To start this migrating motility complex. Okay? Is it clear? Is anything hard in this? And it's very easy, guys. I don't really see anything hard in it. No. So fast review, motility is secreted. This, do you mean the 70% and the 30? Okay, okay. This sentence, it just says إنه نحنا the complex starts from the antrum. Most of the time, it starts from the antrum. 70% it starts. 70% of the times it starts from the antrum. 30% will start from the duodenum. That's all what it's saying. Because if you guys see, this migrating motility complex is something that happens all over our GI. It's not something that only happens in the stomach. And if you see here, guys, we have the antrum, we have the duodenum, we have the jejunum, ileum, and so on. But this migrating motility complex is something that happens all over the GI. They're just saying, no, usually 70% of the time it's initiated in the antrum, but only 30% it can be initiated from the duodenum. So a very, very, very fast recap. Motility is secreted by the duodenum. It will cause the migrating motility complex cycle. The cycle is made up of 3 phases. Those 3 phases happen on a span of 90 to 120 minutes. Phase 1 is the relaxation phase. It makes up 50% of the cycle, and there is no contraction, nothing. It's only relaxation. Phase 2 is where we have the rhythmic contractions. The muscles all together will start to what is it called, to contract and in a certain rhythm, and this makes up 40% of the cycle. And then after rhythmic, we're going to go to the phase 3, which is literally, guys, only 5 to 10 minutes, and it's only 10% of the cycle, and it is very intense peristaltic waves. Okay, is this clear?
Yes, clear.
Yeah, in here he's just showing how it goes. You see, guys, in here the pylori, and it starts from here. Can you repeat the last slide? This slide? Or what?
Before
what
it. Before it. Hey, it's easy. لا تبع ال backward pushing. أيوة
This
هاي.
one?
Yeah.
Can you repeat it بالآن? Like, can I go on?
Yeah, please.
Oh.
Yeah, لأنه أنا يعني استحيت لكم ترجع تشرحي.
لا it's okay. I can repeat it at the end. بس خلينا نخلص بعدين I'll repeat it for you. So guys, in here, what is it called? This just shows how it is. يعني, you know, this MMC, it's like housekeeping. It makes sure everything is going. يعني, you know how basically even if we're not eating, our stomach is still working sometimes. You know, it's still digesting. You know how it takes so much time to digest and so on. So what happens is, you know, he's just showing you, you know, it begins here and it travels all the way down. As I showed you guys, it's literally what's written here, and it begins here and goes all the way down. So it's very easy. And then this slide, everything in this slide, we already covered it. Best we have this, the gastric emptying. So now we talked about how, you see here, we talked a bit about gastric emptying, but we're gonna cover it more in here. And then when we empty the food, okay? So basically the food is digested, and we want to empty it into the duodenum. And we said, guys, you know, only small particles that are less than 2 millimeters are allowed to pass through the pylorus, okay? So what happens is, to allow this to allow the movement of the particles, there has to be a coordination. This coordination usually happens between the antrum, okay? And then it happens between the antrum, the pylorus, and the duodenum. So we have a certain coordination. عندنا certain things have to happen. It's like a pathway, okay? That's why they call it the antro-pyloro-duodenal coordination, because it includes the antrum, the pylorus, and the duodenum. So what happens is, the antrum has to contract, push the food. The pylorus has to open to allow the food to pass in, and the duodenum has to relax. So it takes in all of the food, because the duodenum, guys, is not as, it's not as big as the stomach, مثلا. But the duodenum, it requires relaxation to accommodate in the food. You get me? So again, what happens? We have the antro-pyloro-duodenal coordination. The antrum contracts, the pylorus opens, and the duodenum relaxes so that the food can easily pass through. تمام? Is this clear? It's a very easy slide, guys. بس إذا بدكم أعيد عادي, tell me. I might find it easy, you might guys find it hard. تمام. Repeat the last part. Okay, so the last part is إنه نحنا we want the very smooth movement of the food. So for the food to move from the antrum to the duodenum, we have to have a coordination, okay? This coordination means إنه الantrum has to push. So antrum contracts to push the food. But then the food has to pass through the pylorus. And so the pylorus is closed, the food will not pass, so it's going to be useless. So the pylorus opens and it allows the food in. The duodenum, okay, is smaller in size compared to the antrum. So it has to relax, and it has to relax to allow the food to go in. So it will relax, and then the food goes in, and that's how basically the food will easily pass in. Okay, is it clear now? Yeah. And this like coordination or emptying happens like 3 times per minute. Remember how we said we have 3 peristaltic waves per minute? So if you think of it, it's the same thing. We have 3 contractions per minute that will cause 3 gastric. فهمت علي? Did you get me? I think I guys confused you. Okay, gastric emptying, it doesn't happen all at once. Not all the contents of the stomach will be emptied. It will be emptied in like شوي, like a bit. We're gonna release a little, we're gonna release a little, and so on. So this will happen in the same rate as the peristaltic waves, which is 3 per minute. Okay? Is it clear?
بس نحن كمان قلنا بالpacemaker region we have to make 3 waves per 1 minute.
Yeah, it's the same thing.
So it's the same waves that we're talking about.
Yeah, so basically لأنه نحن our peristaltic waves are 3 per minute, the gastric emptying will occur in spurts. Do we need to know that? Yeah, you have to, guys, know it. 3 waves per minute. 3 peristaltic waves per minute. It's important, especially for the antrum in here. I'm not sure about the duodenum, but for the stomach, no.
سؤال هلا 3 waves per minute. Are we like talking about the arterium, the pylorus, and like are these like the waves that we're talking about? Well,
yes, you have to understand إنه الwaves they travel. It's not gonna عنا هلا مثلا عنا 1 minute بتبلش المينت, the minute just started. So a wave is released. This wave will go all the way and will travel here. Then another wave will release, and you get me? Like it's the same wave, it's just moving on and on and on, and it's getting stronger and stronger and stronger. You get me?
Yeah.
That's how it's just the same all over the stomach. 3 waves per minute. It's just that the strength of the wave will get, the strength of the muscle contraction will increase as we go down. And the gastric emptying command depends on the waves. Hello? Is it clear? Yes. So basically this is like a revision slide, if you can think of it. So after we eat the meal, okay, what enhances gastric emptying? Like what increases the release of food? It's gonna be basically the what is it called? The peristaltic contraction. So the antrum contracting, the pylorus opening, the duodenum relaxing. Okay, so these are all conditions that increase the gastric emptying. For what slows the gastric emptying, basically doing the opposite of what enhances it. ORAD is the stomach, the mouth, sorry. I'm not seeing him. I'm not 100% sure, but yeah, it's like ORAD. Yeah, I'll tell you something. ORAD is basically, wait, عنا ORAD or عنا code. Okay, you see here we have ORAD and caudal. ORAD means the beginning. Caudal means the end. It's like our mouth. You get me? It's like in anatomy and how, like, especially in embryology, how we take, exactly like cranial and caudal. So ORAD is basically the beginning and caudal is the end. So the tone of ORAD region, he means basically, so the food is here, okay? So if the tone increases, as in we have stronger contractions, it will, exactly, the beginning of the stomach or of the tract, no, the stomach itself. Because if you think of it, the food is here, the food is not down. So the beginning of the stomach in here. So if the contraction is the tone is high and the contraction is strong, this will enhance gastric emptying. But if the opposite happens, it will slow down gastric emptying. Okay? Is this clear? It's very easy, guys. And just
so basically in the ORAD region, the upper region,
yeah,
upper region of the stomach.
Yeah. So next, this is, guys, the last idea, which is that the intestinal phase will decrease the stomach emptying. It's just like how you remember how it decreases gastric secretion. It does the same here. It will decrease the gastric motility. So remember the CCK and the secretin that was released because of the fatty acids, and we said no, CCK is because of the fatty acids acting on the eye cell, and the acid that is from the food is going to act on S cells, and they're going to release secretin. And we said both of them, they will activate what? They will activate the vagal nerve, and the vagal nerve will cause the vagal nerve will act on here, and it will cause basically decreased it will, what is it called? It will decrease the tone. It will decrease the peristalsis, so the antrum is not going to contract anymore. The pylorus will constrict, and the duodenum is going to contract. Okay, is this clear? If it's a lot, I can repeat عادي. Just tell me, guys. In here, it's the same idea as the gastric secretion. Yeah, sure. So basically, we said the food is now in here in the duodenum. Okay, the food contains fatty acids, like long chain fatty acids. The food is acidic, like the chyme is acidic because it was exposed to gastric acids, and so basically, when it goes into the duodenum, this fatty acids they will basically they will trigger the eye cells to release CCK, okay? And the acid will trigger the S cells to release secretin. Now, the CCK and the secretin, they will go and they will, like شايفين, you see these are basically, this is the enteric nervous system, okay? It is activated, and the vagal nervous system is activated. So what will happen in here? So because the vagal nerves, the vagal pathway is activated and the enteric nervous system is activated, they know we're activating them to stop gastric emptying. So what happens is that nitric oxide will be released. Guys, do you remember this? Do you remember this? Like, do you remember the pathway? Yeah, it's the same idea, but it's coming from the duodenum. Okay, so what happens is in the duodenum, the release of CCK and secretin will activate, what is it called? And حتى serotonin will activate the vagus nerve and the enteric nervous system to basically activate inhibitory neurons to release nitric oxide and VIP. For nitric oxide, how will it act on the antrum? It will decrease the fundic tone. So basically, it's not going to contract as strong as it used to, and it will decrease the peristalsis. So we're not going to have like the 3 waves per minute. We're going to decrease the peristalsis like maybe to a lower number, or basically it can go down to 0. Okay, and it will actually cause the pylorus to constrict. Okay, so the pylorus constricts, the antrum cannot push the food anymore, and the duodenum كمان it will start having its own peristalsis, and all of this is because of the release of CCK and secretin. Is this clear? Yani all of these are what prevents gastric emptying. Exactly.
So basically,
yeah.
what happens
So-
is that the
Wait 1
in
second. Can
the
I say something? Yeah.
extrogenic yes, sorry, خلاص.
Yeah, but I'll just.
I was just gonna say that in like basically the upper region, the NO and VIP, they like inhibit the constriction, so like they relax it, and the lower region they constrict it more, right?
Yeah. You can
That's
know
it.
it like this. صح. So basically, someone else doesn't decrease in fundic tone means relaxation. So basically, accommodation. Exactly, that's true. So basically, fundic tone is relaxed. So basically, yani if you guys think, we said for emptying to happen, we need to have those. We need to have contraction. We need to have the antropylori duodenal coordination. So the antrum has to contract, the pylorus has to open, and the duodenum has to relax. So to prevent gastric emptying, the opposite has to happen. So what happens is the antrum needs to relax. That's how the nitric oxide in the VIP will cause the relaxation. The tone will decrease. And then the pylorus will close and the duodenum will start its own peristaltic movement. So all of this will prevent the gastric emptying. Is it now clear? And most important thing to know is that CCK is the most important thing that causes this. Why? Because guys, you can see in here the doctor said LCFA is prolonged. هلا نحن fatty acids, they take so much time to get like distributed and to get digested and so on, but they're always exposed to the receptor in here. So that's why they are more strong in bringing about. Can you explain? Yeah, sure. Okay, so let's go step by step. نحن we want to inhibit gastric emptying. Why do we want to inhibit gastric emptying? Because the duodenum has food already. We don't want more food, okay? So what is going on, what is it called? So we don't want food to come. خلاص, we have the food there. So in the duodenum, we're gonna have food that is made up of fatty acids, okay? And we're gonna have basically a lot of acid from the acid secretion. Wasn't the food in the stomach and it was exposed to acid secretion? So we're gonna have a lot of acid, okay? Now the acid and the fat, what will they do? They will stimulate certain cells. So let's take the fatty acid first. Fatty acid, it will stimulate eye cells that are found in the duodenum. هدول eye cells, they're here, okay? Those eye cells, they will see the fatty acid. So they're like, okay, خلاص, I will release CCK. Okay, so CCK is released. And then we have acid. The acid will bind to the S cells in here and it will release secretin. Okay, so now we have CCK and we have secretin. CCK and secretin, they're going to go all the way into the vagus nerve and into the enteric nervous system to cause inhibition of the antrum and of the duodenum and so on. Okay, so it think of it as CCK and secretin. What will they do? They will cause the release of nitric oxide and VIP by the enteric nervous system and the vagal reflex, just like in here. Guys, مثل هون, you see how remember this this pathway, the same pathway literally happens in here. Okay, but it's initiated from the duodenum. Okay, so nitric oxide in VIP is released, so the fundic tone decreases. Okay, so because it decreases, this means the muscle relaxes. So because the muscle relaxes, think of it this way, when we want to empty the stomach, we want the antrum to contract, we want the pylorus to open, as we mentioned here, we want the antrum to contract, we want the pylorus to open and the duodenum to relax, so it accommodates the things. But in here, the opposite is happening. What happens is the nitric oxide, it will cause the antrum to relax, so it will not contract, and the pylorus, okay, it will constrict, and the duodenum will start having its own peristaltic wave because it has the food in it. It needs to move it from the duodenum on. Okay. Someone said increase of fundal tone, meaning increased contraction, stimulates gastric and relaxation habits. Exactly. Literally, this is a summary of fundic tone and relaxation and contraction. What I was saying, no, the main thing that causes the release of nitric oxide in VIP is the CCK. You might ask me why, because basically the long chain fatty acids, the long chain fatty acids are there for a long period of time. They're not gonna get absorbed, they're not gonna get broken down, but that's why they are the main powerful thing. Okay? Tamam? Is this clear? And the other ones are just transients. They happen for a while and then they leave. Tamam? Is it clear,
Yes,
guys?
clear. Yeah.
Tamam. This is the last slide, and it's literally what I just told you guys. شايفين? This is the S cell. This is the I cell. It releases CCK. CCK, what will it do? It will go to the vagus nerve. This is the afferent. This is the efferent. It will stimulate the inhibitory. Okay? This inhibitory will release nitric oxide in VIP. So basically, it will relax. Tamam? Now, LCFA is basically the fatty acids. So fatty acids, they start all of this because they are the reason why CCK is stimulated. Tamam? Is it clear?
Yes.
Do you have any questions? Tamam, perfect.
Can you return the accommodation part, بس?
Accommodation part? You mean in the duodenum?
Yeah.
It's just here it's just an idea and نحن duodenum is نحن we want the gastric emptying to happen, the duodenum has to relax. So when the duodenum relaxes, it accommodates them. But نحن when we want to inhibit gastric emptying because we already have food in here, it will contract, okay? It will have its own peristaltic waves. تمام?
Okay. Thank you.
This is the last slide, guys, and it's a very easy slide. It just says, basically, liquids are like very, 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 very rapid to get emptied from the stomach. And if you can look in here, it takes 2 hours to release this. It's very rapid. You see, a huge amount of food is getting rapidly emptied. And the funny thing to me صراحة is when we have a lot of liquids, as the volume of liquid increases, the emptying is faster. إذا عندنا نحن small amount of liquid, the emptying is not going to be as fast as if we have larger amounts. Okay, this is just things that you guys have to just memorize. And then so this makes sense. Liquid is emptied faster than solid because it's easier to pass. Okay, and then the high fat solid, you see this is high fat, this is low fat. The high fat will take the most time. Why? مو حكينا نحن the fat will decrease the gastric emptying, right? Do you remember this? We said fat is the reason why gastric emptying is slowed down. Yeah, so that's why high fat solid foods are gonna take the most time in basically being emptied from the stomach, followed by low fat solid, and last but not least the normal solid that has no fat. That's it. person with high metabolism, meaning digestion is quite rapid, is it good or bad for the person? Why would it be bad? It depends. If مثلا their metabolism is breaking down their food more than what they eat, يعني مثلا natural collagen takes time, right? Yeah, it does. بس هلا مثلا guys, metabolism is الواحد if the person is breaking or like burning calories more than what he's eating, that's bad. But if he's just burning whatever he eats, then that's good for him. Okay. Exactly. Because fat stimulates Gastric secretion, which releases fundus, so less gastric emptying. صح. Retropulsion takes time, right? Yes. هلا what is it called? I'm gonna explain that slide for one last time, and then if you guys have anything, just ask me. And I think that's it. Okay, for this slide, I'll go over it one last time, and that's it. So, guys, نحنا, when we want to push the food out, okay, the antrum is going to contract. It's going to contract very forcefully. This forceful contraction will push the food, and the pylorus will open. هلا, only the small particles of the food. Is this whole lecture just a bunch of repetitions? Am I just not getting? It is, wallah, it is all repetition. يعني it's just repetition and adding small parts. If you have any question, عادي, ask me. But let me explain this and I'll answer whatever questions you guys have. So basically, those small particles that are less than 2 millimeters, they're going to be pushed out. Yeah? The large particles will not be pushed out. يعني خلاص هلا تخيلوا مثلا a large particle is dislodged in here and it's stuck. So to prevent that, the stomach will contract, but in the opposite way. You get me? It will contract backwards. But then when it contracts forward to push the food, it will contract backward to push the large particles back. And this is called retropulsion. Now, this contraction backward is very strong. It will close the pylorus and it will close the antrum. And it will force the large particles to be stuck in the what is it called? In the body of the stomach, and so that it can get digested into smaller parts. Is this clear?
So basically, in the retropulsion, we open the pylorus and the atrium, not atrium.
No, بالعكس, we close them. Retropulsion, we close them. We shut them closed. So that
after the pylorus is open and we get the food, the small particles to go back. We close the pylorus and then we crush them until they are خلاص small.
Yeah, يعني إحنا we close it to push everything back. You get me? Like it
Yes.
closes and pushes everything back, and it closes so that they're forced to break down. Tamam?
Yes.
Oh. What is it called? That's, guys, يعني if you have any more questions, you can ask me. As I told you, my contact information is in the first slide. Don't be shy to ask me any questions. I know my WhatsApp, my email. If you want to meet in uni, I don't mind. If you want me to explain anything for you in real life, come on, I don't mind. Thank you so much, guys, for attending. I hope I made it easy for you guys. Oh, yeah, that's all. And thank you so much for the host. Karama, thank you so much for